Good morning to everyone. I request all the speakers to mute their audio buttons, please. Once the sessions will start by 10 a.m.
Sir?
Madam,
sir,
please.
I'm having one clarification.
Yeah, please, madam.
Sir, I'm Kausalya. Sir, we have to give the attendance, sir.
என்ன மேடம்?
Attendance எப்படி கொடுக்கணும் சார்?
இந்த chat box இருக்கு மேடம். chat box இருக்கு பாருங்க மேடம். chat boxல உங்க பேரு அட்ரஸ் மட்டும் டைப் பண்ணி அனுப்புங்க மேடம்.
எது chat box இருக்குன்னு சொல்றீங்க?
உங்க போன்ல இருக்கும் பாருங்க மேடம். அந்த நீங்க இந்த
ஓகே ஓகே சரி சொல்லுங்க சார்.
இந்த மெசேஜ் மாதிரி சிம்பிள் இருக்கு.
சரிங்க சார். மெயிலுக்குள்ள போகணுமா அப்படின்னு கேட்குறேன் சார்.
இல்லை மேடம், இந்த ஸ்க்ரீன்லேயே மெசேஜ் மாதிரி ஒரு சிம்பிள் இருக்கும் பாருங்க. மெசேஜ் போட்ட மாதிரி ஒரு சிம்பிள் இருக்கும் பாருங்க, ஒரு ஸ்கொயர் பாக்ஸ்.
சரிங்க சார்.
அதில் டைப் பண்ணுங்க.
ஓகே சார். ஓகே, தேங்க்யூ சார். தேங்க்யூ.
Sir, good morning. Someone else is presenting from the mobile. So could you please ask them to?
Sir, good morning, sir. Sir, good morning, sir.
Morning.
Good morning.
Morning, everyone.
இந்த வாழ்க்கைதான் அழகான வாழ்க்கை. அவர்கள் இருக்கும் வரை இந்த உலகம் நன்றாக இருக்கும். நம்மை சுற்றி என்ற இந்த விவாதத்தை நிறைவு செய்கிறேன். நன்றி, வணக்கம்.
Hello. Hello. Good morning.
Good morning, sir.
Please, all the participants, please close your video.
Video click college.
Jay Prakash.
Sir, good morning, sir.
Good morning. Hello.
Good morning, sir.
கேக்குதா?
கேக்குது சார்.
இப்போ ஓகே, மோர் தென் 99 வந்துட்டான்னு 10 o'clock ஷார்ப்பாக ஸ்டார்ட் பண்ணிடலாம்.
பண்ணிடலாம் சார்.
ஓகே, நம்ம வசீரன் சாரும் வந்துட்டாங்க.
சார் வந்துட்டாங்களா சார்?
ஆமாம், வந்துட்டாங்க.
நல்லது சார்.
ஃபஸ்ட்டு நம்ம சார் நம்ம இவர் எங்கள் ப்ரொஃபஸர் இருக்காங்க.
மதிவானன் சார்.
சார் வந்துட்டாங்களா? ஹலோ. எஸ் சார். எஸ் சார், குட் மார்னிங் சார். கேக்குது சார். எஸ் எஸ். ஓகே, மோர் தென் 99 வந்துட்டாங்க. 10 o'clock. எஸ் சார். we can start sharp at 10 o'clock. Yes. Yes, yes, he is on. நம்ம சார் நம்ம
எங்க டைப் பண்றது மெசேஜ் இல்லையா? சரி.
ப்ரொஃபஸர் இருக்காங்க. மதிவானன் சார் வந்துட்டாங்களா? ஹலோ. எஸ் சார், குட் மார்னிங் சார். கேக்குது சார். எஸ் எஸ். மோர் தென் 99 வந்துட்டாங்க. Participants, please keep off your camera. It will be save your database. Means dear participants, please turn off your camera. Participants, please keep off your camera. Dear participants, please turn off your camera. It will be save your data.
Good morning, sir.
Yeah, good morning.
Hello, good morning, sir.
Yeah, good morning. Please turn off your camera to it can be save your data.
Yeah. Okay.
Yeah, please. And while program is running, you keep mute your microphone also.
Okay, sir. Sir, can't you do from there? Can't you do from your side both audio and video for all participants?
Yeah, it can be shared to all the participants.
No, sir. Audio and video, if you can move from there, it is better.
Why?
No, like Zoom, if you have option, you can mute in single click all the participants' video and audio.
No. It is not available, this option. We can. It will be cause some ethical issues.
Okay, sir.
We don't stop their privacy. Dr. Greener, sir?
Yes, sir.
Yes, sir. Is there any options to mute all the participants except the presenter?
Sir, we can mute everybody and they have to unmute if they want. But when they are joining, if they are not muted, we have to do it manually.
Okay, okay, sir. Okay, okay. Then we will inform in frequent.
Yeah, only thing is when they are joining, there are two options, join now or present. So some one of them are joining through present now rather than join now. So
Yeah.
that's the reason why their screen is getting shared.
Okay.
ഇതിൽ ഇപ്പം കേൾക്കില്ലല്ലോ.
Yes, sir. Sir, good morning, sir.
Good morning.
Yes.
Dr. Selvankumar, are you hearing
Yes,
me?
sir. Yeah, yeah. Yes,
Okay.
hearing.
It is clear?
Yeah, clear, sir. Clear.
Yeah, okay. Thank you.
Yeah, very good response. More than 136 members joining now. Totally more than 280 people are registered so far. It's a very good response from all the scientific peoples among the within our country. Dear participants, please turn off your camera. Dr. Jayaprakash. Jayaprakash.
Sir, please, sir.
கேக்குது. Jayaprakash?
Yes, sir.
Yeah, you turn the slide and it mentioned for the turn of the camera and mute the microphone. It will be keep it in display. Yeah. Yes, very good.
Hello.
Yeah.
JP. Yeah. Dr.
Sir, good morning, sir.
Yes, sir. Good morning.
Very good morning. Yeah. Could you see me? Could you see me?
Yeah, it's coming,
Yeah.
good, sir.
Very good morning. I'm Madhivanan.
Yes. Good morning,
Okay.
sir.
Fine. Yeah. Very good morning. How are you, JP?
Fine, sir. Fine, sir.
Okay. So.
Okay, let's start our program, sir.
Yeah. Yes.
Yeah. So,
But JP, how to operate the PPT?
I'll share, sir.
You will share. Okay. You please share the latest one.
Yes, sir. Yes, sir.
But I have to see in my laptop, no desktop. Can I open and see?
Yeah, you can keep your laptop in front of you, sir, and you can open this.
Yeah, I have a laptop in front. But how to? You will be operating the PPT?
Yes, sir. I am operating, sir. I will operate, sir.
Okay, sir. All right.
morning.
Very good morning.
Yes, let us start the program with welcome address. Good morning, one and all present here. On behalf of Biological Science Forum, we are organizing the e-conference on contagious disease management, a perspective to COVID-19. I welcome Professor Dr. B. Vaseeharan, Professor and Head Department of Animal Health Management, Alagappa University, Karaikudi. Welcome, sir. I welcome Professor N. Madhivanan, Professor and Director, Center for Advanced Studies, Department of Botany, Madras, in University of Madras. Welcome, sir.
Thank you.
I welcome Dr. Girinath Pillai, Nairo Research India, Cochin. Welcome, sir. Next, I welcome Professor Prasad Burra, Professor of Department of Biotechnology, KLEF, Deemed University, Andhra Pradesh. Welcome, sir. And welcome Professor Magima, Assistant Professor, Department of Microbiology, Muthiyammal College of Arts and Science, Rasipuram. Welcome, ma'am.
Thank you, sir.
Yeah, next I welcome, extend my welcome to Dr. V. Naveen, Research Fellow, Mons Biomedicine Discovery Institute, Mons University, Australia. Welcome, sir. Next, I extend my sincere welcome to Dr. Gomadi Arunan, an independent researcher from Chennai. Welcome, ma'am. Next, I extend my sincere welcome to Dr. Niyaz Ahmad, Assistant Professor, Department of Biochemistry, Sacred Heart College, Tirupattur. Welcome, sir.
Thank you.
Next, I welcome Mr. Muhammad Asif, Satyabama Institute of Technology, Chennai. I extend my welcome to Swayam Banerjee, Research Scholar, Environmental Epigenic Lab, University of Calcutta. Welcome, sir. Extend my sincere welcome to Dr. Jnanendra, Assistant Professor, Department of Biotechnology, Yongnam University, South Korea. Welcome, sir. Now we welcome our this coordinator and organizing secretary, Dr. M. Jayaprakash Vel, Head Department of Marine Biotechnology, Amity University. Welcome, sir.
Thank you, sir.
I extend my welcome to all the participants, those who are interestingly registered and keep in online. Welcome you all. Now I invite Professor Dr. B. Vaseeharan, Head and Professor, Department of Animal Health Management, Alagappa University, Karaikudi, to deliver presidential address. Please, sir. Sir, Vaseeharan, sir. Sir.
Yes. Are you here? Now you are...
Yeah. Okay, sir. Please.
Yeah, very good morning, all of you.
Good morning, sir.
And most respected Professor Dr. N. Madhivanan, Professor Dr. T. Selvankumar, and Dr. Jayaprakash Vel, Dr. Greenath Pillai, Dr. Prasad, and Dr. Mahima, Dr. V. Naveen, Dr. Gomadi, Dr. Nivas Ahmed, and Mr. Muhammad Asik, and Dr. Chandra Sandham. A very good morning again, all of you. I am very much interested and I am very happy to participate this e-conference. First of all, I would like to congratulate and give my blessing to the Dr. N. Selvankumar and his team. He is earnestly working to organize such type of conference presently. Presently, you all know worldwide shutdown is going on. You all know that COVID-19 struck the world and we need to prevent and how to manage the disease. We all need to discuss and arrive at solutions. At this juncture, Dr. Selvankumar and his team are doing well. Actually, the participants of all colleges, all universities, all even foreign countries also participated. This is a nice occasion. I am happy to present one among you. And let me brief about the theme of the conference. The conference entitled Contagious Disease Management, a Perspective COVID-19. As we all know that disease management, the prevention and cancer. Hello?
Yeah, okay, sir, please.
Prevention plans we can use to control the disease with probiotics, chemicals, antibiotics, immunostimulants, plant-based products, nanotechnological products, lot of pharmaceutical products. These all products nowadays available with control the disease management, but till now there is no availability of drugs for COVID-19. At this juncture, the conference will pave a way to a very good directions and leading ideas to prevent the COVID-19. Within this, I think the very good experts teams are available in this conference. Dr. Madhivanan, I am very happy Madhivanan sir is here. I think he is a plant specialist. He is talking about the traditional plant-based medicines or biomedicines, how to relate to or how to control COVID or disease management. I wholeheartedly welcome and support you, sir, your plannings and all things.
Thank you.
And my humble request to all the participants, all of them, please, we can remind 3 C's, that is cooperation, collaboration, and celebration. Please all cooperate how to control this disease and collaborate, each one of you, collaborate it. Then finally, after we overcome with this COVID, we can celebrate it. This is my message. And once again, I congratulate the team for conducting the e-conferences. அனைவருக்கும் வணக்கம். இந்த கருத்து பட்டறையானது எல்லோருக்கும் இந்த COVID-19 எவ்வாறு கண்ட்ரோல் பண்ணதுங்கிறத பற்றி நம்ம பேசி ஒரு முடிவு எடுக்கணும். வாய்ப்பு கொடுத்தமைக்கு அனைவருக்கும் நன்றி. முக்கியமாக பயாலஜிக்கல் சயின்ஸ் ஃபோரம் அதனுடைய அட்மின் அண்ட் அதில் இருக்க அனைத்து மெம்பர்களுக்கும் என்னுடைய வணக்கங்களையும் வாழ்த்துக்களையும் கூறி விடைபெறுகிறேன். நன்றி, வணக்கம். Selvankumar?
Yes, sir. Thank you, sir.
It's okay. Thank you.
Okay. Yes. Next, I invite Professor Madhivanan sir for keynote address, please.
Very good morning to each and every one of you who are all linked in the e-conference platform. I appreciate the good effort of the Biological Science Forum for organizing this conference through online. And I thank profusely Professor Vaseeharan Dr. Selvakumar, Dr. Jayaprakash Vel, for having thought of organizing this interesting, important conference, which is deal with the COVID-19. And also I thank for giving me the opportunity to deliver the keynote address. My topic is chosen on the perspective of traditional medicines and plant-derived drugs for the management of COVID-19. I have chosen because of the current trend prevailing in India. JP, can you change the slide?
Yeah, the screen is on live, sir.
It will move on automatically, or I have to move it?
Please indicate, sir. I will share the slides.
Yeah, please change over to next slide. Yeah, I have designed my talk in this fashion, like introduction of COVID-19, the current situation of COVID-19, and some of the suggestive preventive curative measures of this deadly disease. I'm not able to see the slide. Could you please help me, JP? Then traditional medicines and medicinal plants. Why we wanted to include these traditional medicine either for preventive or curative measures of COVID. And then phyto compound as a proven drugs which already been discovered and are being used for various disease control and disorder management. And antiviral activity of medicinal plants. Finally, possible phyto compounds against COVID-19. Next slide, please. Before going to the topic, just I wanted to give an introduction. I'm sure many speakers are going to tell us about the importance of COVID-19 and also its science behind and etc. But just I wanted to start with the introduction so that others can get to know about this deadly disease. This is a novel strain of coronavirus. This is a human coronavirus. First of all, detected in December 19, following an outbreak of disease named as a COVID-19 later, with pneumonia, that is high fever and respiratory complications, the city of Wuhan in China. Initially, it has been named as a 2019 novel coronavirus. Later, it has been renamed as SARS-CoV2 by the Coronavirus Study Group of International Committee of Taxonomy of Viruses. And these coronaviruses, this is not the only virus. We already have SARS and also MERS, and so many viruses are there. They are positive sense single-stranded RNA viruses under the family Coronaviridae and subfamily Coronavirinae. So these are the scientific information about the novel coronavirus. Next slide, please. Next slide, please. JP, can you move on to next slide? Look at the origin of SARS virus, whether it's a SARS-1 or MERS or this novel SARS-CoV-2. The bat is considered as the origin. It has SARS bat is the reservoir for the coronaviruses. This SARS-CoV-2 is started from bat and then pangolin. Then from pangolin, it is transformed to the human being. This is the origin and transmission, how it has occurs. When compared to the SARS-1, again the bat is the common reservoir. From bat, it has gone to a wild pig and then infected a human. Then coming to the MERS, MERS also a coronavirus, the Middle East respiratory syndrome virus, and again bat is the origin of MERS, and then it gone to camel. From camel, it has come to human. So these are the way it is started its origin and is infecting animal, and through animal it has come to human being. This is the general belief of the root of the coronaviruses. So coming to the spread now at currently. Next slide, please. This is the slide I have taken yesterday night. This is the representation how the COVID-19 has spread across the country. It has presented by the WHO, and you can find from the website. And as of now, I mean, the yesterday data, close to 20 lakh people have been infected by this COVID-19 and about close to 1.2 associated deaths. In India, it is about 12,000 and also around somewhere around 200 deaths. So the data I'm presenting, the next slide. Go to the next slide. Coming to the severity of COVID, although COVID-19 has emerged in China, it spread rapidly to about 213 countries. Hence, the WHO, World Health Organization, has declared this as a global health emergency pandemic. So that the magnitude of COVID-19 is so high, and as on 15th April, just now I told, the data, about 19.9 lakhs people have been infected and world over, with a confirmed death of 1.2 lakh people. It's a very, very huge number. Coming to the figures, next slide, please. Again, I have taken from the WHO. The world over, this is the exact figure. But the maximum the infection occurs in United States, though it is started late compared to China. More than 66 lakhs people have been infected, and more than 20,000 people have been died due to this COVID-19. Next to United States is Spain, then Italy in terms of total cases. But if you look at China, though it is originated from China, only 82,000 people have been infected, but only 3,000 plus people have been died. So they are effectively managed to contain the coronavirus. So the number is very less compared to the United States and other European country. If you look at the Indian figure, yesterday figure, it's 11,933 and 392 associated death. So you see the magnitude. Next slide, please, JB. Go to next slide. Hello.
Sir, it's on, sir. Next slide.
Pardon?
Symptoms, sir.
Yeah, mine is working. Yeah, coming to the symptom, again, I have taken from the Australian website. This normal symptom is fever. So probably everyone would have known by this time, because through media, through TV, these symptoms have been keep on informing the public. Normal fever, fatigue, and also respiratory symptom like sneezing, running nose, sore throat, dry cough, and very importantly, shortness of breath, so breath complications. And it's not only affecting the lungs, and it's also affecting the kidney function, and also it is creating problem in intestine, due to which the patient used to get the diarrhea, and again circulatory system also get affected. So the blood cells will be decreased, the white blood cells will be decreased, so that the immunity will be going down in the patients. So therefore, it is a multiple issue. And not only that, it is also creating problem in heart as well as liver. So the infected person is having multiple complications. So therefore, the fatality is taking place. The more importantly, the people with medical complications like heart patients, diabetic patients, or cancer patients, so they are more prone to this particular disease compared to other normal people. So these are some of the symptoms, and many more symptoms probably you will see through the website of WHO or other websites. Coming next slide, please. Next slide, please. Now, the important point is preventive or therapeutic measures. So what are the preventive therapeutic measures so far? we are actually advocating. So this is the new disease, very recently identified, especially in December 2019, just around 4 months back. So therefore, not much research has been done. So therefore, there is no vaccine or direct-acting antiviral drugs are available for the treatment of SARS, that is the virus SARS-CoV2. But the WHO is suggesting to isolate the patients, confirmed patients, or even suspected persons. Therefore, every country now advising social distance. That is the best way to prevent the disease spread. In addition, the WHO also recommending oxygen therapy, fluid therapy, and also antibiotics, because not only the viral infection, it also leads to bacterial infection, because the entire the immune system is going down. Therefore, a lot of bacterial infection is started occurring. So therefore, we need to treat these patients with the antibiotic to contain the bacterial infection. So these are some of the preventive therapeutic measures that have been already on the round, and this all based on the previous information. They gathered the information through SARS, the one, as well as the MERS. So those information now they are trying to employ here. Next slide, please. But what is happening actually? Next slide, please, JP. So what are the drugs and how we can address these preventive therapeutic measures? Next slide, please. Hello. Go to next slide. No, previous slide. Previous slide. Yeah. Coming to the drugs. So already everybody knows about the hydroxychloroquine. This is not the drug for the coronaviruses, but it has been discovered from a plant. The chloroquine is discovered from plant Cinchona. It was advocated for malaria. So, but now they have found out this hydroxychloroquine, if you administered with azithromycin, this antibiotic, the COVID intensity is come down. So this is the one. And another antiviral drugs like remdesivir, lopinavir, ritonavir alone, or in combination with interferon B. So now they are also recommending it. In addition, there is a humanized monoclonal antibody, tocilizumab. This actually now they are recommending for the treatment. In addition, the convalescent plasma, so the plasma therapy. So everybody now talks about the plasma therapy. Even yesterday, I came through the media, and India also now planning to go for plasma therapy. But this plasma therapy is not new for the coronavirus. Actually, it has tried during the Spain flu period. So because the plasma therapy was very effective, that's what they found out. And China also, they tried this plasma therapy, and this also now in treatmental measures. Next. So these are the, but still the point is not all these therapies are proven for the control of the COVID-19. So it's all only suggested because nothing is available. Therefore, they are suggesting these therapeutic measures. But what we can think of for controlling this COVID, here I wanted to throw some light on the alternative control measures. The thing is now everybody knows, whomsoever are watching the TV or the media, especially in India, many now practitioners are Indian medical practitioners, especially they are practicing Ayurveda, Unani, Siddha, or homeopathy. They are talking about the Indian traditional systems of medicine for the preventive or treatment of various diseases, like viral diseases. It's already there. Previous slide, please. But despite a lot of speculation and debates, actually a lot of debates also was on the TV for the use of these Indian traditional medicines. But in spite of that, and the common people, they started using it. Even many the NGOs as well as the hospitals, Siddha hospitals, they have started distributing some of the Indian traditional systems of medicine for the treatment of the COVID-19 at present. We are seeing this is a common occurrence nowadays. But the bottom line is that there is no scientific studies on these traditional medicines for the treatment of coronaviruses, whether human coronaviruses in general or SARS-CoV2, the specific virus. There is no scientific evidences, but it's all only anecdotal evidence. So their personal experience they are sharing, and they are advocating to go for the plant-based Indian traditional systems of medicine. Next slide, please. Some of the medicines, actually, some of the preparations, they are not medicines, they are preparation like Nilavembu Kudineer. It's already, even government of Tamil Nadu, they were recommended during the swine flu as well as the dengue fever epidemics, use of Nilavembu Kudineer as well as the papali leaf extract. So Nilavembu Kudineer now people have started using, and Kapasura Kudineer. It's actually, everybody talks about now Kapasura Kudineer, which is effective against this coronavirus, Corona-19. And also the Adudoda Kudineer and some other preparation like Sudarsana Suranam, Thiruchengode Suranam. These are some of the traditional preparations, and now people started using without even recommendation of the medical practitioner. People themselves are started using it because everybody is in the confusion state, how to treat, how to get rid of this corona, how to prevent the corona. So therefore, they are finding ways and means how to protect themselves. So these are the preparations already in the use. Next slide, please. This is not story, this is the reality. So we are seeing. So why herbal medicines? What is behind it? So herbal therapy is well established in most of the ethnic cultures, including India, and have become a way of life. If you look at the world, almost 80% of the people, especially in the rural areas, they are practicing the herbal medicine for their health benefits. And not only in the rural areas, even the developed countries like US and Europe, almost around 30%, 25 to 30%, doctor prescribed medicines contain the plant component. So therefore, there is a growing interest all over the world to use of herbal medicines. And now it's order of the day. Everybody started working on herbal medicine to find out molecules or find out the preparations to tackle various diseases and disorders. Next slide, please. Next slide, please. Oh, how they are actually initially started selecting the plants. Actually, they are not looking at the medicinal aspects, but while searching food, the ancient people, they found out some foods had specific properties of relieving or eliminating certain diseases and maintaining the good health. So they found out certain the food based plants have shown the health benefits. So therefore, from there, the search of medicinal plants have been started. Next slide, please. So I am presenting this. You know what are these ingredients? It's very, very common. Every kitchen we used to have this. It is very famous in South India. It is known as next slide, please. Thiruchengode Suranam. Thiruchengode is nothing but the pepper and also the typically we used to call in Tamil, as well as the ginger. Ginger officinalis, Piper nicarum, Piper longum. It's a combination. If you mix equal proportion and make it a powder and put it in hot water and drink, and people believe that it cures many diseases and we will get rid of many other problems. So it is a normal practice. Though these plants are food ingredient, but they are very famous for health benefit. Coming to next slide, please. Next slide, please. Now the Choranam is available commercially. You can buy. You need not even prepare yourself. You can buy and you use it. But the preparation is very cheap. You can buy these ingredients from shop and you can prepare yourself for your health benefit. Next slide, please. And this another preparation, again, three plant component and very famous, especially is in Ayurveda. The previous one is Siddha, this is Ayurveda. This is known as the next slide, please. Tirupala Choranam. Tirupala Choranam, it came from Terminalia cupula, Terminalia bellarica, and Embolica officinalis. All three equal proportion these fruits, and you can mix it and make a powder and you can use it, and it's very famous in Ayurveda, and it's also having a lot of health benefits. Next slide, please. Again, you can see this Embolica officinalis. Every day we are consuming as a pickle, as even raw also we can consume, and the commercial products are available. Why I'm presenting these things? These are there in Indian systems of medicine. Nothing is new. So our ancient people, our forefathers, they found out these medicinal properties of the plants, and they try to use it in their health benefit measures. So therefore, we can think of these kind of plants to find out some health benefits, not only for coronavirus or any other virus or any other health benefits. Next slide, please. Next slide, please. Coming to some phytocompounds as a proven drugs. So I'm showing only the preparation. Already scientists have been discovered some phytocompounds. Those phytocompounds have been proven as a drug. It is available commercially. I am presenting some blockbuster molecules for your information. Next slide, please. Next slide, please, quickly. You know the quinine. Quinine came from Cinchona officinalis, from the bark. They isolated, and it was the first discovered medicine for the malaria. Even today, we are using it. So the hydroxychloroquine came from this quinine only. It's a derivative of quinine. So it is from plant. It's a blockbuster molecule. Today we are using. Even today we are using. Next slide, please. Next slide, please. Yeah, you know pretty well about the Catharanthus roseus. Is known for anti-cancer property. Vincristine, vinblastine, it's a blockbuster molecule and identified, and they are using as an anti-cancer drug, commercially available. Next slide, please. So time is up, so therefore quickly I'm presenting. The another molecule called camptothecin came from Camptothecin acuminata. It is actually known as a happy tree or cancer tree. So scientists from America, they discovered, even Indian scientists also involved in this discovery. So they identified this in the advanced clinical stage. They isolated this compound from the bark of this tree. So therefore, there is a potential. We can look the plants as a source of drugs. These are a proven drug. Next slide, please. So another interesting drug I'm presenting, Taxol. So everybody knows now, Taxol is a very important molecule, blockbuster molecule for cancer. It came from Taxus brevifolia. The same scientists, the two scientists previously I presented about the camptothecin. Same scientists have been discovered again from tree bark. They identified, and it is a highly commercialized product in the market. It is available for the treatment of cancer. Next slide, please. Next slide, please. And another compound called vernolepine from Vernonia, and this anti-tumor drugs is it is also in the market available. Next slide, please. Can you? Okay. So I think it's pretty interesting to show this particular compound called metformin. Metformin again came from a plant. It's like a common weed, Calanga officinalis. So they discovered, and metformin again is blockbuster molecule. Even today, globally, so every country is using metformin left and right, especially for the control of the diabetics. So another very important molecule. And next slide, please. Next slide, please. Podophyllum peltatum. So coming here, this is the compound, podophyllum toxin, and for the treatment of human papillomavirus. So this is also available commercially now. They obtained from this particular plant. Next. Next. Significantly, Philander samaras. So it is Philander samaras is a Indian traditional medicinal system, especially in Siddha. People used to give the preparation of Philander samaras as a churanam or something for the treatment of jaundice. Jaundice, the general belief is cannot be treated through allopathy. So people mostly depending on the traditional medicine, especially Siddha. So the Philandin is active compound. They are already identified, and preparations are available commercially, and we can make use. Even today, people are in the villages. They will go to the traditional healer. So they used to give these preparations for the patient, and patient will be get cured from the jaundice. So next slide, please. The jaundice again, it's a viral disease. A very interesting observation. This is Artemia annua. This is a plant, a very common plant in Himalayas, especially in the temperate region. This scientist has discovered by her name is Tu Yu, basically from China, and she has identified artemisinin. Again, this is a very interesting malarial drug for which she has been awarded Nobel Prize along with two more scientists. One is from Japan, by name Satoshi Omura. Another scientist from UK by name William Campbell. So for the discovery of ivermectin, another important compound for the treatment of filariasis disease. And this lady discovered Nobel laureate artemisinin, for which she has been awarded Nobel Prize in the 2015 for medicine. This commercially available, the artemisinin. So a very path-breaking research outcome from the plant. Next. So again, I told, so the COVID-19, again, why I'm presenting here, this hydroxychloroquine, it came from the quinine from Syncona officinalis. Therefore, there is a possibility of identifying some novel compound from the plant source for the treatment of coronaviruses, especially the COVID virus also. So I am just presenting some of the information, especially deals with the viral diseases as well as other diseases, as well as some of the phyto compound which can be utilized for the treatment of COVID-19. So in the coming slides, next slide please. You can see here I listed some of the indicative plants. The list is not exclusive, very, very indicative. There are many more plants have been already reported. They have antiviral activity, starting from Rhinacanthus onwards to Verbascum. So they are effective against herpes simplex virus, HIV virus, influenza virus, adenovirus, something like that. So therefore, there is the possibility to identify some active medicinal plant and with active molecules which may have the solution for the cure of COVID-19. So therefore, I urge the participant, young participant, probably you can take up this kind of research to work on the medicinal plants to find out some novel compound for the treatment of the deadly disease COVID-19. So next slide, please. Next slide, please. This is the compound. Previously, I was presenting one of the plants, and these are the phyto compound which has been isolated, purified, and identified. Chupulagic acid, that is from chupula for the control of herpes simplex virus, and hibomanin from Philandus, urinaria again for herpes simplex virus, coumarin from calophyllum for the HIV, human immunodeficiency virus, and chalcones from Cleriaceae for influenza, xanthones again for influenza. Likewise, there are informations are available for the use of the phyto compound as a compound, not a whole extract, as a compound already purified and identified, and they can be used for the treatment of viral diseases. Not only just its information, and some of the compound have been already developed as a drugs. So coming to the next slides. Go to the next slide. Very interestingly, I wanted to present this information. This is a plant, very common plant in Tamil also. It is known as a sweet root or lucorice, or in Tamil it is called as Adimaduram. This is a root, it is very sweet. If you taste it, it's very sweet. It is a root of Glyceraceae glabra. This is a common plant. It is a part of Indian systems of medicine, not only Indian system of medicine, in Chinese also. Chinese traditional system, they use this lucorice. And interestingly, one publication in the year 2003, Pilcher. So he has published lucorice may tackle SARS virus. And when the SARS-1 came in 2002, and 2003 they published in Nature. So he has recommended this compound glycyrrhizin can be used for the tackle SARS virus in 2003. Very interesting observation. SARS-1, it's the same family, the COVID also. So therefore, we can think this kind of plant compound for the treatment of the COVID-19. Not only the Pilcher, and again, Sinartil et al. So again, 2003, they published this paper in Lancet, very interesting journal, Nature and Lancet. They published glycyrrhizin, an active component of lucorice roots and replication associated coronavirus. So in which they have recommended this glycyrrhizin can be used for the control of SARS virus. So Therefore, there is information already available. So therefore, from there, we can move on to find out some phytocompounds from antiviral plants so that we can tackle this SARS also through the traditional systems of medicine, or find out active molecules, and these active molecule can be researched further and can be developed into drug for the COVID-19. Next slide, please. Next slide, please. So in addition, very recently, 2020, Chen and Tu, they published, actually this is not the peer-reviewed paper, but it has been published in online. So they have suggested potential phytocompounds against SARS. So they are proposed based on the previous information. One Baicalin, it's an active compound from Scutellaria species. There are many Scutellaria species were able to produce the Baicalin as a secondary metabolite, and this can be used. And another compound, Scutellarin, again from Scutellaria species. And then one more compound from lemons and sweet limes, and also another compound, nicotinine from Glycine max, and also the glycyrrhizin from Glyceria glabra or Glyceria, another species. These compounds, they have done bioinformatics studies using the COVID pathogen, and based on the bioinformatics information, they suggested this compound can be a potential compound for the treatment of COVID-19. So therefore, there are informations are available, so we can try. Plants are there, and plant-based compounds are there. The compound informations are available, and therefore there is a possibility we can really work further on these plant or plant-based drugs to find out the effectiveness against the COVID-19, that's SARS-2. And here I wanted to emphasize this COVID-19 is not a rare phenomena. If you look at the history, there are many diseases, many epidemics, many pandemics were there, and through which several millions of people have been died. And every century we are facing this problem, and this is not a new problem. And in this century, in our lifetime, we are facing this issue. Therefore, we need not to get panic out of this. And of course, now we have scientific tools to find out what is the pathogen quickly. And for your information, within just 4 months of time, hundreds of papers have been published in peer-reviewed journals. And it shows that a lot of research is going on around the world. The scientists are trying to find out vaccines. They're trying to find out the drugs for the treatment of prevention or treatment of the COVID-19. So therefore, we need not to panic. It's a very common phenomena. So this anyway we can tackle with the support of the scientific tools. So with this, I summarize, especially the plant-based traditional medicines, identification of potent traditional medicine and medicinal plants against COVID, human coronaviruses could be path-breaking steps towards controlling the viral respiratory infection. So therefore, we can really think of these medicinal plants as well as traditional medicines. But research has to be done, because anecdotal information will not help us. We need to do scientific validation. So identification of antiviral phytocompounds from selected medicinal plants could be potential source as lead molecules, which already I presented through the publications of Chen and Tu, for the development of preventive or therapeutic drugs. And mechanism of action. So we need to do, though we have information, anecdotal information, we don't know how they are acting. So therefore, mechanism of action of traditional medicines and phytocompounds from medicinal plants would provide more knowledge and understanding for their scientific validation against various viral diseases, more specifically coronavirus, especially the COVID-19. So therefore, there is a lot of scope in this area. So I urge and I invite the young scientists to join in this area to work on traditional medicine or medicinal plants to find out a very, very vital solution for the COVID-19. So with this, I thank profusely the organizers, especially Dr. Jayaprakash Vail, Dr. Selvankumar, Professor Vaseeharan, for giving me the opportunity. Thank you. Thank you, one and all. Thanks all the participants.
Thank you, sir. Thank you very much for your nice presentation and motivation to all the scientific community, sir. Your experience and your expertise, it will be fruitful for our scientific community. Thank you very much, sir. Now I invite our program organizing secretary, Dr. Jaya Prakash Vel.
Sir, I think, sir, if you can ask any few questions, if there are any questions from members.
Thank you, Dr. Selvankumar and Jay Prakash. Well, if any questions, probably I can happy to answer.
It's a time for
I have one doubt, sir.
Yeah, okay.
Hello, sir. I am Dr. Mahendra from Kalisuri College, Sivagasi, sir.
Welcome, sir.
Sir, actually, first of all, I thank to you. You are delivered wonderful lecture, sir. It is useful for us for further research. I have one doubt, sir. Actually, the coronavirus came from bird, sir.
Coronavirus, actually, it has come from Wuhan state.
Okay, sir. Actually, animal,
China.
it come from vowel.
Yeah, it has come from bat, vowel, yes. That's what they say,
sir.
yeah.
Okay, correct, sir. But some bad researchers said
Till yesterday.
Okay, sir. Actually, some bad researchers said.
Yeah. Actually, one more information, very recent information, I'll tell you. Till yesterday, I was thinking that it has come from bat. It was there in a lot of websites, and today they are saying it is not come from bat. It seems it's a new information for me.
Yes, correct, sir. Correct.
But till yesterday, the belief was from bat.
Yeah, it's absolutely correct, sir.
Yes.
Sir, actually, some bad researchers said, and actually, the 2018, some researcher like ICMR collect the bad blood groups, sir. Bad blood. They are identified the blood, sir. Actually, some they are traced some virus disease in bad blood. Actually, that virus and now the coronavirus is not synced. They're totally different, sir. Actually.
Yeah. Actually, bats are reservoir for many viruses. That's what there is a lot of literature that are saying. Not only for this COVID, even MERS, even SARS-1, that was the reservoir. That's what they say. But not only they have a coronavirus, they also have many other viruses.
Oh, okay, sir. Okay, thank you for your clarification, sir. Your presentation is definitely useful for my further research, sir. Thank you so much, sir.
Thank you, Dr. Mahendra. Thank you.
Oh, thank you, sir. Thank you.
Okay, thanks.
Hello.
Comments. Yes, please.
Hello.
Yeah.
Hello.
Yeah, I'm hearing you.
I'm Dr. Raghunath Badigal from Park College of Engineering. Sir, there are reports that it can also come through the stool when it is contaminated with the water. Is it true?
Yes, sir. That's what recently people are telling. It is also excreted through stools. Yes.
So there is a possibility.
Yeah, there is a possibility. Yes.
Oh.
Like.
Okay, sir.
Yeah.
Nice presentation. Good.
Thank you, sir.
It is named as bat passage.
Sir, good morning, sir. Hello.
Yes, please.
Sir, this is Srinandini from Kaidamnath Government College, sir. Sir, yesterday I read that is frequently getting muted. So, how the drugs can be worked, sir?
Pardon?
The virus is getting mutated.
Okay.
Yesterday I read an article. It showed that the virus the coronavirus in India is getting mutated. So, in the mutation, how the drugs can work, sir?
Drugs.
Yes, sir. If you are finding a drug for the
particular, this virus is getting mutated. People are believing that China strain and the US strain is different from Indian strain. That's what people are telling.
Yes, sir.
But a lot of information is coming in. So how it is working, the plant-based drugs? For example, there is the ACE, this angiotensin converting enzyme 2, which is the receptor for the viral binding. So some of the peptides of plants and some of the plant compound can block the ACE receptor as inhibitors. They are known as the ACE2 inhibitors. So if you find out this kind of inhibitor, definitely these plant extract or plant compound can prevent the viral spread. So that's what already reports are available. It's not a new information. It's already reports are available. There are peps they identified even from common food crops like wheat or barley. So they identified this kind of peptides.
Okay, sir. Okay, sir. Yeah. Thank you so much for the excellent presentation. Thank you, sir.
Thank you, ma'am. Thank you, ma'am.
Okay.
Any other questions from the audience?
Hello.
Hello.
Thank you, sir. Thank you for your nice presentation and your clarification to our research community vendors' queries. And now, shall we go for next session, sir?
Yeah, thank you, Dr. Selvankumar.
Yeah, thank you, sir. Now I invite this organizing secretary, Dr. J. Prakash Vel. Please give some regulations and instruction to the presenters as well as members to operate this video conferencing, as well as a brief idea about this theme of the conference, please.
Yes, sir. So very good morning to all scientific fraternity. I'm Dr. Jayaprakash Vel, head of the Department of Marine Biotechnology, Amateur University, Chennai. And I'm happy to associate with this team as an organizing member for the conference of contagious disease management use with the perspective.
Google இதுல வாங்க.
Hello.
எல்லாரும் அதுல அதுல வரல. ஃபோன் பண்ணாங்களா யாராவது? ஃபோன் பண்ணாங்களா? சரி.
Hello. Jay Prakash. Hello, Jay Prakash. Hello, Jay Prakash. Hello.
JP, Jay Prakash Vel.
Yeah, sorry, sir. There is a power cut in my area, so I now connected through mobile.
Okay, fine. Yeah.
So sorry for the inconvenience. So this, I think this conference has been, the idea of this conference has been aired by Professor Selvankumar last week only. We have a WhatsApp group called the Biological Sciences, and we share many useful information through this forum. So he has proposed an idea of having a e-conference on because of this lockdown period. He wanted to utilize the services of all senior people and to bring it the knowledge to the young community. He has proposed this idea, and we have formed a small group among ourselves. Dr. Mahendra from Korea and Dr. Giridhar from Kerala. We have joined together with the able support as well as guidance of Dr. Selvankumar. We could able to organize this conference. So there were many difficulties in organizing the technical aspects of the conference as well as we have, but all other our hurdles has become nothing when we see the response. Actually, we had initially more than 200 response, and now you can see more than 190 participants are in the session. That gives immense happy for us to organize this event. Actually, we are on behalf of the committee, we are very much satisfied. So while we discuss about the theme of the seminar, we initially thought that it would be a general topic, but upon discussion, we found that due to this COVID-19 outbreak, let us focus our conference on COVID-19 with special reference to its disease management. So we have framed some technical sessions, and we contacted, we opened the idea to all the people, and the resource person have volunteered themselves by giving their titles and other information, and we could able to frame the technical session in such a way that there are about 7 technical sessions that have been organized today, starting from the traditional knowledge. That's what Dr. Madhivanan has given a wonderful lecture now, and there are some speakers who will be discussing about the historical aspects of the evolution of coronavirus from SARS to MERS and to novel coronavirus 2. And also there are some presentations about the structural aspects of the viruses as well as the information from the structural studies to design as well as to develop the vaccines as well as other testing protocols, there are lectures are there, and we have also lectures on the treatment aspects, like the clinical aspects of the COVID-19 has also peoples are also presenting, and we were very much happy. We are very much happy that from various parts of the world, people have joined together to bring the different stream of knowledge and present it over here. And most of the technical support has been rendered by Dr. Giridhar from Kerala, and we are very much happy for that one. So this is the background of the conference, and with reference to the theme, and I think we have shared all the speakers' titles in advance, and we will be delivering one by one during the session. So with reference to this welcome session, we would like to formally introduce, there is a small slip in the protocol, we would like to formally introduce the people who are present. Firstly, I would like to introduce about Dr. Vaseeharan. He is the professor and head in the Department as well as Animal Health and Management. Professor Vaseeharan has completed his BSc Zoology and MSc Zoology from Madurai University, Madurai Kamaraj University, and he has completed his PhD in Zoology in 2002 at the University of Madras. He is having more than 15 years of teaching experience and 20 years of research experience. And his area of specialization is biomaterials and biotechnology in animal health. He has completed so far 8 PhD guidance and ongoing 8. And 7 MPhils have been completed under his guidance. He has also guided more than 50 PhD students. He has published, he is a very good voracious publisher. He has published more than 200 research articles where he is H-index process 30 with the citations more than 3,000 in number. And he has attended more than 50 conferences, both international as well as national area. And he has published 4 books. He has handled 13 research projects and 3 are ongoing. The amount amounts to be more multiples of crores. He has organized 15 events. So that is our patron, Professor Dr. B. Vaseeharan, and we were very much happy to have Professor Vaseeharan in the inaugural session. And we also had the first keynote address by Professor N. Madhivanan. He is Professor and Director Head in the Center for Advanced Studies in Botany, University of Madras, Chennai. He is having more than 30 years of experience, research experience in the field of bioactive metabolites, especially for the management of plant and animal diseases. He has completed his BSc from Kannur Government Arts College as well as MSc from Annamalai University, and he has done his PhD at the University of Madras in the area of plant pathology. With specialization in plant pathology. Later, he worked in Nagarjuna Agricultural Research Institute as a scientist, and he has moved into University of Madras in 2002 and taken charge as a lecturer in the Department of Botany. And later, he has elevated to several positions like associate professor and professor. And 2015, he has taken charge as director of Center for Advanced Studies in Botany at the University of Madras. He has also received so much of award, including the very prestigious TANSA Scientist Award, and we were very much happy to have Professor Madhivanan in this forum. So, I think all of you are able to hear me.
Yeah,
right. Sir. Yeah. So that is about the speakers we had so far, and I would like to propose some of the important instructions to the participants. I think you must be experienced that there is a problem in sharing the screen. That's what we have requested, and we are requesting all the participants please not to share any of your screens. Please not to share your screens so that the speakers alone can share their screens. The respective speakers will be sharing the screens, and that will be visible to all the people. Without any hindrance, the speakers can present their presentation. This is the number one instruction what the cooperation we are expecting from the audience. And other thing is, you please, all of you, please mute your audio as well as video. That will reduce the data consumption as well as there will not be any disturbances during the presentation, because background noise coming in and it is propagating a huge noise over here in the bigger forum like this. So I request all learned participants to switch off their mic as well as video So whenever you have, we will be opening sessions for discussions. At the end of presentation, we will open for discussion, and we will we have plenty of time. Please don't bother. We have plenty of time. We will be giving time for elaborate discussions. So we will, once you have any points to raise, then only you please switch on your mic as well as switch on your video. Now, I request every one of you not to share your screens, as well as not to switch on your audio as well as video. Without by muting your audio and video, you are still in the presentation only, and you can very well witness the presentation. So with this very short this thing, I hand over the session to Dr. Selvankumar, and maybe we can move on to the next technical session.
Yes, sir. Thank you, Jayaprakash. Hello.
Yes, sir. Yes, sir.
Yeah.
Sir.
Yes. Tell me.
I think next speaker we can invite, sir. Dr.
I invited Dr. Thiruchengode. Please introduce his profile to the participants.
There is a small issue, sir. I have a power cut over here, and I would have printout.
Okay.
I think.
Okay. Shall I do?
We can invite him directly, sir. We can invite him directly.
Okay. And later we can.
Yes, sir. Later, sir.
Next, I invited Dr. Greenath Pillai. He is an eminent personality for operating and distribution of some bioinformatic tools. We are one of the collaborator for purchased some softwares like CSAR FluxX from his association. So his presentation will fruitful for our research community for developing some new drugs by using some tools like bioinformatics and pharmacokinetics tools. Now I invite Dr. Greenath Pillai for his presentation. Welcome, sir.
Hi, sir. Good morning. Thank you for the invitation.
Yes. Please introduce a few points about yourself.
Sure. No problem. Are you able to see my screen?
Okay, what's the problem?
Are you able to see my screen?
Yeah, of course.
No, I am not. We are not able to see your face or your screen.
No.
Screen. Yeah, PowerPoint.
Screen is there, sir.
Yeah,
okay,
we are able to see.
Great. You have to just click on my name probably. Then whoever not able to see, you will be able to see my screen. Okay, great. So,
Dr. Greenath.
Yes, sir.
Please allow me to take you.
Hello, sir. Hello.
I'll take you. Hello.
Hello, sir. Yes, sir. Please.
I'll take the privilege to introduce you to the audience, sir.
Okay.
So,
fine.
Dr. Greenath Pillai is the director and research scientist at Zastra Innovations, Bengaluru, and formerly he is a Marie Curie Research Fellow at Molcode Limited, Estonia. He carried out his PhD at University of Florida with Professor Kartritsky as well as Professor Carlson at University of Tartu. He holds more than 40 research papers in peer-reviewed international journals. More importantly, he is having 2 US patents to his credit. And he also authored one book, co-authored a book. Currently, he is guiding PhD students in the area of aging as well as antiviral therapies. Apart from the scientific position, he is also a mentor at IIT Kottayam and VIT as well as the Government of India's Niti Aayog ATL schools. So with this brief introduction, I request Dr. Greenath Pillai, Director, Zastra Innovations, to present his talk on SARS, MERS, and now COVID-2. Please, sir.
Yes.
Thank you so much, sir, for the nice introduction. Of course, I don't want to take too much time there. And thank you all. It is an immense response from everybody, and there are many people joining also. And I really thank the previous speaker who was giving a good insights about the traditional medicines, preventive measures, and a little bit of history. So that was really nice. And now I wanted to talk a little bit more about our history. If you see the arena of SARS, MERS, of course, Middle East respiratory syndrome related diseases, and now it's COV2. We need to understand there are some relations here. And if you look at the sequences also, there is similarity also. So on a computational biology perspective, we can see some relationship here. But when coming to diagnosis and treatment, it's a bit challenging on whatever the perspectives and assumptions that we are keeping in our mind. So we need to, as many of the speaker and others have already hinted, we have to do a lot of research as well as come up with solid evidences. First of all, we wanted to understand the terms and names. It's very important because many of us get confused. As already the previous speaker, Dr. Manivannan, has already mentioned about ICTV, CSG. I'm not going on to all those names. But the virus species for MERS is Middle East Respiratory Syndrome related coronavirus. And for SARS and CoV2, it is a severe acute respiratory syndrome related coronavirus. And they all are named as MERS-CoV, SARS-CoV, and SARS-CoV2. When you're calling it the diseases, as it is already called as MERS and SARS, but for disease name, here it is called as COVID-19. And the year it has evolved is 2012, 2003, and 2019. So that's where you see the difference in what it has come. And we also have to understand what is the terminologies that to be used. So understanding this, I'm going for the next slide to explain about, of course, already we have had a confusion here until yesterday, or until we were always thinking about bats. Now there is a different assumptions. The source might be from different other animal or a bird. So we have to have a better confirmatory to make that claim. As of WHO, this is the data. And as per the publications which I have cited at the bottom of the slide. So the virus has come from bat, most of the CoV related virus, but the intermediate between the human and the bat are different animals. One is the civet cat, then camel, and pangolins. So MERS and SARS, at the date of March 3rd, WHO was saying that SARS-CoV2 was having only 3.8% of a fatality rate when compared to MERS and SARS. They were of around 9%, 9.8% and 34.8%. But as of today, I'm sure the fatality rate is not anymore 3.8%. It would have tremendously increased because it's a global pandemic situation.
Dr. Giridhar, sorry to interrupt. Dr. Giridhar, sorry to interrupt. Somebody else is sharing the screen.
Please don't share your screen, please. Yeah, okay.
So so understanding this, we need to think of the origin of coronavirus. As of today, we will keep it as bat anyway. But understanding, as I told you, SARS-CoV and SARS-CoV2 has some relationship because both have outspread during winter. One is in winter 2002, other is winter.
Slide is not visible, sir, please.
Oh, sorry. Oh, okay. I hope now it is visible. Right? Is it visible now, sir?
Yes, sir. Yes, sir.
Visible, sir. We have seen, sir.
Okay. Great.
Again, somebody is presenting. Radhika. Madam Radhika, please stop sharing the screen. Madam Radhika, please stop sharing the screen.
Yeah, I can see that. Okay.
Sir, please, you don't give a full screen, sir, so that only you'll be knowing what's happening. Somebody else seeing the screen, you will be knowing. Please be in the view only. And we don't see your screen now.
Oh, okay. Yeah, because the other person is viewing, that's why. Oh, okay. I hope now you are able to see.
Yes, sir. Yes, sir. Please.
Great. Thank you so much, sir. Yeah, this happens in online platform. so it's okay. All of you, please bear with us. Now, coming to SARS and the COVID-2, we can see that the relationship with what the emergence criteria both came in winter, and the cases are more, and the deaths are also more in COVID-2 now, and the countries affected is also now more compared to COVID. And the receptor in both the human receptor, as already Dr. Manivan mentioned, it is ACE2. But coming to the clinical approved inhibitor available, there are many available, but we have to do a lot of clinical trials. So drug repurposing is going on. That is the best solution for an immediate answer for the question of inhibition. Because other all new novel compounds, of course, it takes time for it to get into the market. So drug repurposing is the best solution right now available with every researcher and the pharma industry. And they are already started and doing it. Neutralizing efficiency by SARS convalescent sera. That's what plasmid already Dr. Manivanan has mentioned about it. Yes, sir?
Yeah, please go ahead.
Okay. So SARS-CoV-2 cell entry depends on AC2 and the other, and it is also blocked by a clinically proven protease inhibitor. This is what ultimately we are looking for. So whatever we are doing to do drug discovery or any drug design, we are looking for the need of the hour. Getting it after 2 years and 7 years might be for the next pandemic we are prepared. And even yesterday I read the clinical trials that is going ahead also, only by 2021 we'll be ready with a drug. That is the situation now. But as already WHO, ICMR, and many other CDC and many other disease control and councils, they have already suggested a combination of drugs which is support treatment or support medicine rather than a confirmed medicine available. Of course, we are talking about hydroxychloroquine, erythromycin, and many azithromycin and many others. But they are all supportive drugs, which is for each step of what is the kind of replication, to stop the replication, to stop the interaction with the human receptor. That is where what we are talking about. I didn't put that slide here because I don't want to mislead any of them to know the right name of the drugs through these slides. Anyway, to understand coronavirus into the spotlight, I have this is a picture that is taken from a journal, Viruses 2019 March, where they have published. So they were talking about CoV entry and MERS. So we are comparing about SARS-CoV and MERS-CoV. If you see here, the MERS, the receptor is DPP4, which is again on the humans. And when you're coming to SARS-CoV, the receptor is ACE2. So for SARS-CoV and SARS-CoV-2, the receptors are same, but on the MERS, it is different case. But again, if you think that can I use the same similar drugs that's being used for SARS-CoV, still we don't have any drugs as such defined. That is the reason why we are still going behind SARS-CoV-2. So it enters the target cells through an endosomal pathway, and the spike protein, that is S is for spike. The spike protein of SARS bind to cellular receptor angiotensin converting enzyme 2, and the spike protein of MERS binds to cellular receptor dipeptide peptidase, that is DPP4. So these are the two receptors where it is responsible, one for CoV and the other for MERS. Okay, so and if I go to this, back to this picture, you can see here M, E, and S. So these are the different levels. What it is being mentioned here: E is for envelope, M is for membrane, and N is for nucleoplacid. So this gives a better understanding about what is the mode of action and what is the kind of replication happens when the virus gets affected with the human cell and when it gets replication. This is a picture that I have taken from WHO as well as from scientificanimations.com to understand the real picture of the SARS-CoV-2. As you can see, this particular region is the spike glycoprotein. That's why we are talking a lot about it to understand, okay, that is where the ACE2 is being getting interacted and then pulled into the cell. But we are also talking about membrane protein, that is here, and then hemagglutinin esterase, that is here. And then there are envelope, membrane envelope, then RNA and nucleated proteins and envelope proteins. So now there is a lot of research going not only on spike protein, they are going towards glycans, membrane proteins, envelope proteins also. And that is where the research is moving forward for a better understanding. Now, coming to the genome sequences, whatever is available for CoV2, we have to understand whatever that is being reported. Many of the genomic experts are saying that they are not still complete because the assemblies to be made. So genome sequencing, so we need to have access to raw sequence and do proper assembly of the sequences to further carry out our research. So it is very important to understand on the sequence level if there is any bioinformatics or computational biology participants here. Now, these are the key protein targets. Of course, there was a participant who was asking about mutations. Of course, it is already reported that it is of slow mutation, and when it is getting spread out from different population to different population, you can see different strains or variants because they are still yet making understanding of mutations. So far, whatever the protein targets that is being crystallized, there are no mutations found or reported yet, but there are co-crystal ligands available. If you see, there are many of the drug discovery studies are happening on 6YB7. 6YB7 is the protein target they are working on, and as well as on 6M03. These are the two main protein, as well as 6VW1. 6VW1 contains the AC2 receptor of the human as well as the nCoV. So it is nCoV was the earlier name that was given by from China, and then later WHO renamed it to COVID-19 anyway. So nCoV chimeric receptor binding domain is what you can have it from 6VW1. But 6YB7 and 6M03 just contains the protease of SARS-CoV2. So these PDB IDs, protein data bank IDs, these proteins can be used for your studies on molecular docking studies or even for molecular dynamic studies. So this is just a snapshot that I have taken from diamond.ac.uk, where they have done extensive studies on 6YB7 on covalent as well as on non-covalent. There are key amino acids. So you have to have a better understanding before doing any sort of drug discovery studies that these kinds of studies requires understanding of which is the key amino acid. So here you can see histidine 163, glutamine 166 motifs are some of the important amino acids in this particular motif. Coming to the other catalytic side, of course, cysteine 145 is something very, very important. So they are non-covalent hits over catalytic cysteine 145. So if you ask me what are the key amino acids, one is cysteine 145, then histidine 41, and the other one which I showed you like histidine 163 and glutamine 166. So these are the key amino acids that you have to consider when you are considering the protein 6YB7, which is a protease protein of CoV2. Now, how are these therapeutics could be discovered? We need to have a different mechanism. We cannot do everything together. As our keynote speaker, we already mentioned that there should be collaboration, cooperation, and it should be. So we need to do target discovery, target identification, lead optimization, ADME and toxicity, animal testing, and process optimization. So just doing a single job is not going to make us to give the results come on. Oh, sorry, someone is, okay. So we have to have a better understanding of where we have to target. What is the biomolecular target that is tied to disease? Now, I don't think that all these areas can be covered by a single person or even by a single area of research. It is a multidisciplinary area. So everyone should join their hands together to come up with a particular research agenda and to do it in a proper way. So animal testing is also something very important. So for that, you have to understand we need to have BCL3 facility, laboratory facility for doing any kind of antiviral testing. So BCL3 facility is not everywhere. There are very limited laboratories, including academics, who have this facility. But I know in most cases we don't have BCL3, but at least BCL2 will be there. This is a standardization required for biosafety when you're working with viruses and other organisms or species. So, and the process optimization is also very important because we want to design the mechanism for manufacturing at a scale of therapeutics. That is something that is to the next stage. But up to animal testing, this is within the academic scope. If you have facility, if you have a good scope of collaboration, all these things can be achieved. So this is something that we have to always overthink and do it, because we are not looking for just a purpose of publication. We wanted to scale it. We want to get it in the market or patent it. Now everybody talks about open science. No problem with open science. The only thing that you have to be careful is in open science is that you share the data, everything with the public and without keeping it secret. So that is very important, that even though you patent or even though you want to scale it and bring it to the market, keep your science open so that everybody can comment on your data and they can review it. Now, why there is no vaccine yet? The therapeutics can be very dangerous. Swine flu influenza vaccine in 1976 may have caused Guillain-Barré syndrome. So that is what there are after effects of the vaccines. So the therapeutics can be really very dangerous. As we already know from news that people have already started vaccine clinical trials. We have to wait for that. No other choice. It's not an immediate solution. Scaling up manufacturing is a bit hard, and best hope the short term is from repurposing the existing drugs, which I already stated that the only hope for short term for fight against this pandemic is the repurposing the existing compounds. There are earlier success stories that repurposed compounds of existing drugs have been used for other diseases. I'm not going much into those details. Now I wanted to give you more details about the resources. So I know there are plenty of researchers, PhD scholars in this participants. So this might be very useful for you. You can take a screenshot of this slide. Now the first one, the Kaggle, it gives you the complete literature data set of what all things have been published so far on COVID-19 or COV2. So you will get the full details from there. Antiviral compound dataset, around 50,000 compounds from CAS database. They have compiled it based on antiviral properties as well as based on similarity search. They came up and compiled it. Of course, you need to have a good amount of computing power in order to do molecular docking or any kind of drug designing projects using this 50,000 molecules in that CAS database. D-Shaw is a company which works on molecular dynamics. So they have uploaded around close to 10 GB. I know it's huge, almost 10 GB of molecular dynamics trajectories of the COV2 protein as well as with ACE2. So unfortunately, you need not to do any of these calculations. It's already being done sophisticatedly in computers, and they have already loaded the data. Your job is to only download them, make your analysis and interpretations. So if you say that I don't have good computers to do, you don't require, because already the data, everything is being uploaded as an open science initiative. That's what I said. Everybody did. Of course, they want to get cited, but it is open science. They share the data, whatever they got. Then drug repurpose. Accelera is a company in Hyderabad, and they have come up with a database only for COVID-19 or COV2. Sorry. I think my presentation is visible to everybody. Someone has reported it is not visible.
Sir, it is visible, sir. We are seeing it.
Okay, great. Sir, whoever is not able to see my slides, just click on my name, then it will be visible. Okay. So drug repurposing data is also available from Accelera. That means whatever existing drugs that is being approved, that they have made a selection and they have listed few of them. So you can use that data also for your drug discovery studies. Now coming to genome resources, every country when they are getting this COVID being affected by their population or citizens, they try to genome sequences. India also has genome sequence already too, and there are more coming up because they are already doing it. And you will get the genome resources from IGIB. IGIB is an institute in India, in Delhi. So you will get the resources from there. Then, as I already told you, we need to do the assembling of the sequences when you have access to the raw sequences. The computational protocols are also given in open. Anybody can use it, nothing secret. They're given every step, which tool is required, how you have to do it. Because from person to person, on your views, it might change actually. So it is recommended that you can use those resources so that IJB is also providing these kinds of resources. The second is, and the last one is protein interaction. Protein interaction means whatever proteins I have shown you in the previous slide, BioGrid has already generated what are the possible interactions for specific compounds or compound data set or even repurposed ones, how they are interacting with these proteins. So that way, you need not to redo the docking and all because the data is already available. What we need is the human expertise on interpreting the data. That is what we require. So there are a lot of resources of SARS-CoV-2 is available. Now, only thing now we require is how to fight again. We need to have different perspectives on mind mapping and also brainstorm it and see how we can actually do things much better. Now, there are also resources for research funding, as many of you are thinking, okay, I have idea, but I don't have money. What will I do? As you know already, BIRAC is already has called for, I think end of this month, you have the deadline. Then DST, SERB also, Bill and Melinda Gates Foundation, European Union Commission, European Innovation Council. Don't think that is only for Europe. No, it's worldwide. Anybody can participate. Then in India, we have few startup missions like from the government initiative, Kerala Startup Mission, Startup India, which is from Delhi, then Karnataka Startup, Jharkhand government. I don't know anything with Tamil Nadu, whether TNSTC or Tamil Nadu Startup Mission is doing something. Please inform me if there is something there, so I'll update my slides. And now, if you think that I don't have softwares, of course, there are free softwares available. You can always use them. But if you think that you wanted to sophisticated softwares, of course, many of the sophisticated software commercial tools, they're giving the free licenses for non-profit and academic COVID-19 projects. You cannot use these projects for any commercial consultancy work. Of course, you can use it completely for free for COVID projects for non-profit and academic. Now, it is not only the software provided by us from all over the world, and they're giving you minimum from 3 months to 6 months of licenses. So you can make use of this if you're working on COVID-19 projects. Now, we are also forming a consortium for an EU project, European Union project. Yesterday only, we have submitted all the documents, and we are forming a consortium. And the details you could find in this particular website, that is github.com, GiriBio COVID-19. So we will be, it is completely open science and not for profit. So that's why I'm keeping it very open. Anybody can come in, but keep in mind, we are looking for specific expertise who understands about chemical biology, metabolomics, pharmacologist, toxicologist, then computational biologist and medicinal chemist. Because here it is more of a drug discovery. We have to design and come up with derivatives from the existing molecules for as an enrichment factor and how we can improve the efficacy. That is what we are looking for this particular project. Of course, you can contact me, or you can go to this particular link and you can you will get more details there. So I really like to thank you for your patience, and I would like to thank our superheroes. Superheroes are doctors, policemen, all the delivery people, and officials who are helping us to keep safe us and home. And I also thank all the organizers of this e-conference, and as well as inviting me. Even though I am not an expert of virology or even on antiviral studies, recently we have got into it, and an emerging researcher on antiviral research. Earlier, I had worked on HIV type 1 on NNRTs, and after that, I'm just going through COVID-19. So my area focused more on a mechanistic driven drug discovery. Let it be any area, because mechanistic driven is very important, because without understanding that, drug discovery doesn't make any sense. I also thank my PhD student, Ambli Uni. She also helped me with this project to carry out. And Vijay Kamath, who is not a, what to say, hardcore researcher, but his questions are actually helping us to find answers, because provoking questions helps in research to for us to think, okay, this could be that way, what could be the hypothesis. So he's a person who puts questions on us and helps us to motivate us to do next level of research. And also Sajida Lulu from VIT and all the group of researchers we are discussing with this project. And thank you all for your patience. And I'm happy to take any questions if you have. You can unmute yourself and I'll be happy to answer.
Yeah, sir. Good morning, sir. I'm Dr. Mahendra from Kalisuri College, sir, Sivagasi.
Yeah.
Sir, I have one basic doubt, sir. Sir,
Sure.
actually, I'm a botanist, not a virologist, sir.
Okay.
Actually, my doubt is, what is the major difference between SARS, MERS, and now coronavirus, sir? And which one is highly dangerous, sir? What is the reason?
I cannot say highly dangerous. All are dangerous only. The thing is, COVID-19. People are still exploring it, and not much has been explored now. There are what to say, limitations of access to data. I don't want to very specifically mention it, but otherwise, SARS, MERS, so SARS-CoV-1 or CoV and CoV-2 are having good similarity. Around 99.1% similarity is there on the sequence. But that difference of 0.9 is making it a big issue here, making the difficulty in researchers to find a proper solution. So that 0.9 difference is making the problem. But between the MERS and CoV, coronaviruses, as I told you, the receptor where they are interacting is completely different. So they have a good amount of difference. But coming to CoV and CoV2, there is a similarity of 99.1%.
Okay, sir. Okay, sir.
So all are equally dangerous. So we have to see. The only thing which I can see is stop traveling, so that at least this will not get spread to others. And staying at home means that makes the sense just one sentence: stop traveling. That's the only thing. Even outside the country, within the city, within the town limit, whatever it is, stop traveling.
Okay, sir. So again, one question, sir.
Yeah.
Actually, why we have struggled to discover the medicine against coronavirus, sir? What is the reason?
Can you please repeat that question, please?
Sir, actually, why we have struggled to discover medicine against coronavirus?
Hello? Okay. See, the struggle is not because of discovering a medicine. The struggle is that as per the standards of FDA and CDC, we have to test it. Clinical trials has to be done, and that takes some time. It is not immediate. So today, if you give to a human like a candidate a testing medicine, and tomorrow you cannot get the result. They have to actually monitor for 2 more days. So the clinical trial is actually getting delayed. Now there are more than 12 companies who are testing on RNAs, then drugs, as well as vaccines. So already it is much forward.
Okay, sir. Sir, no, another one question, sir. Actually, there are some village traditional peoples, actually, not to Marthavar, they are discovered, they have some medicinal plants against the coronavirus, but our Indian government not approval for us. What is your opinion, sir?
See, there is a reason. For coronavirus, there is no medicine. Okay, and maybe people are testing it. In the Natu Marundu, whatever Ayurveda, whatever Siddha, they are very good medicines to increase our immunity, to rejuvenate ourselves, to have a better energy. All those things is good. Okay, in good sense, I'm telling. So I cannot say that that might be the reason why government do not want to make a misconception or misunderstanding to the community. Okay, there is a drug for coronavirus. No, they don't want. They already said from Ayush Ministry to boost your immunity. Please follow this.
Okay, sir. Okay.
And they're all traditional medicine.
Okay, sir. Thank you for your clarification.
If you have better immunity, you're safe.
Oh,
To some
okay, sir.
extent.
Okay.
Disclaimer.
Okay, sir. Okay, sir. Thank you for your
No
clarification.
problem. Any other, sir?
Sir, I have one question.
Yep.
Actually, this is
Hello, sir. Hello.
Yeah, please go ahead with your question, madam.
yeah, okay. So I have one question, sir. RNA virus.
Hello, sir. Could you give some examples for docking software, sir?
Sure, sir. Sure, sir. She's about to ask some question. Can you please complete that, madam? Madam, not able to hear you. Yeah.
This may be uses are you able to understand?
Madam, you can type the question. I'm not able to hear you.
Problem,
So
sir.
let me answer the question for the one sir has asked me. Sir, see, Autodoc is a software that you can use it for free, no charges. Then there is HairDoc and PatchDoc, which is available for free. Now, when I told you for COVID-19, there are softwares available for free. That is CSAR for COVID research, for post-era submissions. They are giving free licenses. Then I know that Discovery Studio is also giving free licenses. Then who else? Then there are FarmSlera, who gives their licenses for pharmacophore modeling. Then StarDrop from Optibrium, they are also giving free licenses on pharmacokinetics, ADME properties and all. So there are plenty of softwares. Now, when you're specifically asking for docking, I would recommend you to do several iterations on different tools, at least 3 or 4, and then see which one is really giving something closer to the experimental data and pick that as your software. I cannot say A or B or C, this is the best software, because A might be good for some set of biological like targets, B might be good for others. So it depends upon the algorithms that you're choosing. So I cannot name that Autodoc is good for everything. No, Autodoc has its own limitations. So once we know what are the limitations, then we will be able to choose the best one. So I cannot say one is good for everything. No, never. I hope I answered that question. So is there any work done on RDRP inhibitors or RNA technology? Yes, there are a lot of research is being done, and even commercial companies are working on RNA technologies. Whether silver and gold nanoparticles based drugs can be used as COVID-19 disease. Madam or sir, that silver and gold nanoparticles, we really don't know, because I heard that zinc is a good for containing or stopping the interaction, zinc actually, and then azithromycin, of course. So you need to have a better wet lab experiments to confirm that question, so I cannot answer that. RNA virus will react with WBC. My question is the mechanism of any drug reaction with this virus. You're right, because people are also thinking, also understood the hemolysis of RBC. That's why oxygen limitations are there. Of course, the lung infections are also taking place. So yeah, it might react. We are not sure. So any other questions?
Yes, sir.
Yeah.
Sir, for rapid screening test kit, why we are depending on other countries, sir?
Madam, a rapid test, the first rapid test kit came from China actually.
Yeah,
and,
sir.
Yeah, and they are costing like $2.2 or $2.1 per kit. And so I don't know, but then later in India, now we have all the kits available locally, and there are plenty of tests happening. So now we are not importing, as per my understanding. Earlier it was, but now we have kits. But the problem here is we don't have the sufficient amount, so we have to import. Yeah.
Okay, sir. Thank you, sir. Thank you so much, sir.
People having autoimmune diseases are prone to get attacked by COVID-19. Madam, this is something that is similar to what to say, herd immunity, because we expect the plasmid, the plasma, all those related convalescent serum treatment, all those things are depending on that. So it's too early to comment on those things, because I am also learning like you when I hear from media. If there is no other question, I'll pass on to the organizers so they can continue with the next speakers. Okay, I have a doubt. For example, fish does not have ACE2 receptor, which is an entry point for binding and causing disease COVID-19. If a positive COVID-19 patient touches the fish, the other healthy patient touches the same place. In this case, will it be transferred? Madam, already there was a paper published on what material, how far this particular virus can stay active and live. So for example, copper 4 hours, on steel 7 hours, and on plastics, I think again 7 hours, and on some other material. So your question is on fish. If it is, the question is it within the water or outside? Is it a dead fish? We really don't know. But again, as it is already said, there are at least 4 hours, 7 hours to 12 hours on different materials. It can be alive. So there are chances that if the fish is being touched by a new person, if it is within that 4 hours or 2 hours, I don't know the hours time frame for fish, they might get affected. What is the difference between CoV-2 and COVID-19? CoV-2 is the coronavirus category name. COVID-19 is the disease name. That's what was my first slide to explain the terminology.
Okay. Okay, sir.
There a possibility for coronavirus to spread through dead bodies? Sorry, madam, I really don't know. Okay, thank you so much. I think we are done with the questions. I'm passing on to Selvakumar, sir. Yeah.
Yes. Thank you very much, Dr. Girinath Pillai. Dr. Jayaprakash.
Yes, sir. Yes, sir.
Now I invite our next professor, Dr. Burra VLS Prasad. Please introduce himself to the.
Yes, sir. Very good afternoon. Good morning to all of you. As you see, as we are seeing in the screen, we are very much happy. We are very much happy to invite Dr. BLSP Prasad. That KL University, Vaseeharan, speaking about the COVID-19 pandemic and initial review. So, is a structural biologist, a software developer, and a biotheoretician.
Excuse me. Audio is not proper. Please correct the audio.
Okay, sir. Okay. Okay, sir. Is it audible, sir, now?
You block the video, then you connect the audio.
Okay.
It will be better.
Yeah, now it's better. Thank you.
Right. So, Professor is an experimental structural biologist, software developer, and a biotheoretician. More importantly, Professor Prasad is a first-generation biotechnopreneur starting bioinformatic products in India. He holds a PhD degree in structural bioinformatics and protein crystallography from the most famous institution in India, that is Molecular Biophysics Unit, Indian Institute of Science, Bangalore, India. He is having a vast postdoctoral experience at the Burnham Institute for Medical Research, University of California, San Diego, USA. He is having a glorious 5 years of industry experience in the bioinformatics industry. Presently, he is in the teaching field at KL University. He is having more than 15 years of teaching experience. Professor is having now 2 ongoing projects funded by DST as well as ICMR. Professor has been guiding so many research scholars in the frontal areas of crystallography, biophysics, as well as software development for various biological activities. So now with this short introduction, on behalf of the organizing committee, I request Dr. Prasad.
Professor Prasad, sir. Hello. Hello.
Sir, I think we have to invite Professor, sir.
Yes, yes. Okay.
Yeah, welcome, Professor.
Hello.
Yes, sir. Please open your PowerPoint presentation. That's enough. Yeah.
Is it fine now?
Yeah, fine. Somebody is disturbing. Please, Sheila Krishnan, please stop your sharing. Sheila Krishnan. Sir, you start, sir, please.
Okay, so thank you, Dr. Selvankumar.
Yes, thank you, sir.
And it is, in a way, I should say, it is my first presentation online, invited talk. So, but I'm glad most of the things have been already.
Sir, your slide is not presented. Please, once again, you share, sir, please.
It is visible now?
No, sir. Sheila Krishnan, please stop your presentation. Sheila Krishnan. Sheila Krishnan, please stop your presentation.
I am sharing actually my screen.
No, sir. Somebody is yes, sir. You are. Somebody is disturbing. Sheila Krishnan. Kavi Karunya, please don't disturbing presentation. Kavi Karunya. Sir, now you share, sir, please. Sir?
Yeah.
Yeah. You share your slide, PPT. Yeah. Now it is going correct.
Yeah, good.
Okay.
Your slide is not shared. Now it is presenting. It is on technical issue, I think. You can.
So basically, can I just go on?
Yes, sir. Please, sir. Yes.
Yeah, thank you, this Biological Science Forum, for inviting me. And it's a very relevant topic as of now during this time point. That is contagious disease management perspectives to COVID-19. And I'm very happy that the earlier speakers have summed up most of the molecular aspects of the current scenario across the globe. So I will take the next 20 minutes to 25 minutes on basically overview of this, as the title says, COVID-19 pandemic, an initial review. So what is going to be covered? I thought of covering these aspects about SARS-CoV2, about COVID-19, taken to contain COVID-19 pandemic.
Schedule. Okay.
Am I okay?
Yes, sir. Please proceed,
Okay.
sir. Please.
So the available COVID data. So I will be talking about, because currently I am a member of the Technical Advisory Committee of AP government to understand what is how the patients are getting infected, and pre-symptomatic, post-symptomatic, basically we are asymptomatic. All these things, how they are how it is influencing the total infectivity rate, exposure rate, susceptibility rates, all these things. This is what currently I am trying to analyze the data, and we are also trying to look at the current modeling aspects that are going on. So in that way, I am happy that the data which I am going to present and the post or the measures to be taken, that is the contagious disease management measures, that also I will be touch basing and that's how I will conclude this particular talk. I think it is going to complement whatever has been covered till now. I am happy about that, and thanks once again. Let us go to the about SARS-CoV, and as the first speaker has already talked about the origin, history, structure, I'll just give the overview of this. And here is a phylogenetic tree where the blue dots, blue letters are related to human. And as it is seen here, the outbreak of the disease probably started with single or multiple bat transmission from wet markets in Wuhan. Everybody knows now that the crazy animal game and meat shopping goes on there, and it has already started again. So that is one thing which is troubling again, concerning for us, because it may again, maybe Wuhan is not susceptible anymore, but it may spread to others. So the earlier it was found in 2003, and the next day MERS was in 2012, and this has been covered already. So I will go ahead with the next one, and this has been covered by, I think, Dr. Pillai also, that the bat, and recent news that it may not be bat. That also is one more theory that is going on around. Okay, so let us see, and this is structure of SARS-CoV is also has been talked about, and the genome size here is around 29 to 32 kilobase pairs. That is 29 to 32,000 base pairs, out of which we can expect around 29 products, in a way, 29 products. And most of these products, we can divide it into either vaccine development targets or drug development targets. So basically, this is what's happening. And the most important thing already told are the M protein, S protein, E protein, and N protein. That is envelope protein, spike protein, S, membrane protein, and nucleocapsid protein. And recently as 2 days back or 3 days back, we have RNA-dependent RNA polymerase structure also has been solved. And as you can see, there are two proteases, papain-like protease and main protease. These are called 3CL Pro is another protease, which is, these two are the proteases which actually activate the virus genome structure, genomes, basically genes, to make it viable and infective in a human or in a host. So these are working, or they work on the ORF1AB gene, which produces around 16 predicted non-structural proteins. So in total, it will be around 29 proteins, which are our targets to contain, from a molecular biology point of view, the COVID-19 aspect. So that takes care of this. And as you can see here very clearly, how spike protein and ACE2 are interacting with each other to get into the system, host system, and then RNA, how it is released. And this has been already talked about, and how it is also a very similar replication cycle that is followed by AIDS virus also. During my PhD, I worked on aspartic protease, that is HIV protease is one of them. So at that time, I had a good study about HIV cycle also as well, HIV-1 especially. So let us move forward in a way. So one of the one of the speakers has told that not much genome variations is there, but yes, these genome variations are very important. Considering the drug discovery which we are planning to do, or which we are trying to approach from a discovery point of view, so this particular phylogenetic network analysis SARS-CoV, this has come only just March 17th, March 30th, it has been published. So in that also it has been told that there are subtle differences, but these differences are going to influence the drug discovery as well as designing aspects of the proteases, which is of my current research, which is ongoing research. So I am basically working on vaccines as well, and so let us move forward. So let us come to the understanding about the disease. As just now it has been clarified, when we say a SARS-CoV2 means it is a virus name, and COVID means coronavirus disease 19. Okay, so we talk about and these symptoms, everything has been in the media, and it has been covered very well by the governments across the world. And I just picked up few things here, which is nothing but the symptoms. That is how the symptoms will be. Basically, you have a dry cough, and if you have simultaneously high fever and a sore throat, and also a difficulty in breathing. The special thing which most of the people are concentrating on is the breathing difficulty. The acute breathing difficulty, once we get, we should be very careful and immediately approach the doctors. That is what the major thing about this COVID-19 is. The onset of COVID, as you can see, the incubation period is around 5.2 days, and the outbreak or the death can happen from 6 to 41 days based on the severity of the infection. Okay, and the severity is dependent on the immunity of that particular individual as well, and the community effect. That means how many people are around who got affected with this. So basically, we go back to our own hygiene we have to maintain. So basically, it is dependent on the age of the patient, and I will show that in the next few slides during patient survey, which we got some data where age also plays a very important role in this. But the status of the patient immune system to identify the modes of transmissions. Fecal, as last speaker has discussed, fecal and urine samples also should be tested to exclude the potential alternative route. It has been published on 26th February about the feces also contributing to the transmission. So let us be very careful about this. So what are the different modes of transmission? That is airborne and personal contact, contaminated objects, and mass gathering. So this is one of the major ways. And if you see, in a more scientific term, the key points are respiratory transmission, aerosol transmission, contact transmission. So health, our WHO also has given very clear rules and recommendations and guidelines to be very careful with respect to all these three different types of transmissions. For example, respiratory transmission mainly transmitted between people through respiratory droplets, and these droplets are nothing but which contain the mucus which hosts the virus particles. When symptomatic people sneeze or cough, large respiratory droplets can travel fairly far, and it has been told up to 8 hours. Up to 3 hours, virus can be in viable condition in the air itself. So that is another important thing which we have to remember. Another complicating factor is that at least 25% of the people who are transmitting the virus may be asymptomatic. That means they don't show any symptoms at the point. Okay, so this has been also communicated. And mode of contact transmission still a mystery. Outbreaks of COVID-19 on cruise ships pose a risk for rapid spread of disease beyond the voyage. So this is another article showing that every way of transmission has to be halted till we try to avoid this type of contamination or mode of transmission. We have to stop mode of transmission in every possible way, and that is one reason why there is one slide here which shows difference between, generally this is a common questions we get, what is the difference between cold, flu, allergies and COVID-19. As you can see, most of the things can happen, can be potential, can be present, all these things. So we are yet to get the data. So from a data perspective, not everything is very clear or rosy about the COVID-19 infectivity. We are still to get a lot of numbers before we conclude most of the things. Now coming to the diagnostics, how at all these diagnostics are happening. That is what we are supposed to now understand about this. So there are three methods, especially diagnostics, that is PCR based methods and serological tests, and diagnosis, that is serological methods and lateral flow assay methods and rapid in-clinic antigen tissues testing methods and this blood-based immunoassay kits. So these are the 4 types of methods which have been in the media and in the news because they are the only ways in which we are trying to diagnose positive versus negative. However, we should be very careful because as I tell you in the next few slides, why WHO does not recommend the other methods except for PCR based methods. So PCR itself is basically it is a genetic information, presence of antigen in the form of an RNA. The test should not be interpreted as response of the host immune system. So we are not looking at whether the body immune system has responded to the antigen. It just indicates that the presence of virus. So this is a very subtle information which we should understand. The test is good indicator of who is infected and who should be isolated or quarantined. For that information, PCR tests are very important and isolation. So PCR provides the important information about the locality, regional spread of a disease. Nasopharyngeal swabs are being used for this test and 30% false negatives. So you can imagine that 80 to 90% specificity of detecting the virus. We are not yet there to confirm that whatever tests we are doing is absolutely right. Okay, so we should be little a pinch of salt should be understood when we are hearing the news from everywhere.
Excuse me, sir, please share your slide. Here somebody disturbed. Please share it once again. Yeah.
I will share it again.
Yeah, please. Yes, sir.
Okay.
Yeah, okay. Thank you, sir.
So is it on the same slide which I stopped just now?
Yeah, about COVID-19 diagnostic test.
PCR.
Yeah. It's a PCR based.
Yeah, exactly the same slide. You can proceed, please.
Yes, okay, thank you. Thank you. So basically what I am trying to say here is PCR is the alternative which we have right now, but still we are far away from accurately saying or absolute authority to say that the person is infected. Maybe it is there. So let us go to the next serological, that is antibody based diagnostics. So detect the presence of antibodies against SARS-CoV2. The test should be interpreted as response of the host immune system to the viral presence. As I told you, PCR is response to the antigen, and serological methods, that is antibody-based methods should be interpreted as response to the host immune system. But the only problem here is, as said in the next point, the test is good indicator of what proportion of population is infected, and hence gives an opportunity to call off social distancing and lockdown. So it will not talk about who is infected and who should be isolated or quarantined, because antibodies are generated after a week or 2. Generally, it is considered that once infected, the antibodies will be generated after at least 10 days. So that means we are getting the antibodies after 10 days. So by this time, the virus might have left the body as well, in the form of when it might have contacted many people. So basically, tests also provide information about herd community. Just now, the earlier speaker was talking about herd community. So this particular serological tests, which again help in decision making, whether to call off or continue social distancing restrictions and other lockdown measures. Unlike swabs, blood samples are used in serological tests. As I tried here to distinguish, we have a swab method, which takes the RNA or DNA as a antigenic material for identifying the infectivity or presence of the virus. The blood samples are used to detect the antibodies generated against the virus. So if we get the immunity period after
Sorry for the interruption. Please put back your slides. Somebody has
Mr. Sakti Salwan, please don't disturb the presentation.
Sir, please
Sakti
Put
Silvan,
your slide.
Yes, sir, I'm doing that one.
please stop your presentation.
Sakti
Who is
Selvan
not
is
interrupting this presentation? Sakti Silvan. Hello. Yeah, sir, please.
Am I okay?
Yeah, okay, sir. You are okay.
So if we get the immunity period after COVID-19 infection, we can develop plasma therapy. So this is very good news. I'm very happy to share this, that plasma therapy may be not a permanent solution, but yes, it is going to be the immediate solution that is possible to control or contain this COVID infectivity across the different people. So either vaccine technology, which will take a long time, I will come back to that. But plasma therapy is one thing. So what is plasma therapy? We will come back to that again. So now the lateral flow, these are commercially available Sona nanotech, and the most important thing here is that this test directly detects the covariance virus. I'm sorry, coronavirus 2. It is a proprietary technology, and it is based on a biomarker, very typical biomarker, a specific biomarker. The sample can be sweat. This is the advantage of this particular technology, as they are claiming that you can use the sweat, urine, blood, saliva, serum, and any other body fluids. It is very interesting to observe, and also it seems it takes 5 to 15 minutes only for detection, and less than $50. So I think this is available in next 2 to 4 weeks. I think I will say that it will be available in next 2 to 3 weeks, because this is only a 3 days back slideshow. I mean information. Now, in the same way, there is a rapid in-clinic antigen testing. The Bosch has developed the technology. It had earlier developed Vivaltic analysis device, and now it has improvised further, and it is based on swab antigen test. It detects not only SARS-CoV, coronavirus 2, but also it has another 8 respiratory viruses in the device. So it is the one of the first fully automated molecular diagnostics device. So it can deliver the results test in 2 to 2.5 hours. The device will be again available in Germany first, and April, and later it will be expanded to the rest of the world. So that's what they are doing. So my actual work has been to understand how the diagnostics go, and what is the patient samples, and how they are being, whether we can believe this or not. So I think it is going to complement whatever the talk has been given till now. So BioRed also is coming up with its own detection kit. So this is a summary table of the same, and I think I have covered almost most of in the earlier slides. So let us get back to this. So only 34. After reading all the things, what we can conclude is only 34 to 85% are accurate. All these diagnostic, and there are many false positives are high. So how much to believe? So this demands for accurate, reliable diagnostics test. And if you see in the next few slides, I will be showing you that more than 41 different diagnostic tests are under screening and consideration by the FDA for approval. So let us get back to one. WHO recommendations, the point of care immunodiagnostic test recommendations from WHO. So rapid diagnostic tests based on antigen detection are not recommended. RDT, rapid diagnostic tests on post antibody detection, are not recommended. Only molecular PCR based respiratory tract samples are recommended method of identification and laboratory confirmation of COVID. So this has been advice given on the use of point care immunodiagnostic tests for COVID by World Health Organization. Just to tell that whatever you hear in the media or whatever you experience around you may not be absolutely correct. There is still a chance of recovery. That's what I would like to here emphasize. So in this perspective, there is some disturbance somewhere. Is it okay? Am I?
Sir, it is okay, sir. Please go ahead, sir.
Okay, thank you. So after understanding what are the diagnostics which are available and the molecular biology and epidemiology aspects, now let us see what the governments or the steps taken to contain COVID-19, this pandemic. So I have divided these things into immediate, short-term and long-term efforts. So immediate preventing steps are hand washing, hygiene, do's and don'ts, and let us see what are they. So not much, it is about cleanliness, personal hygiene and safety. This is what is indicated in these cartoons which are shared here, which are shown here. Wash your hands often and wear a face mask. Avoid contact with sick people. Always cover your cough or sneeze. Very interesting, and 20 minutes hand wash is what is prescribed, and the governments have taken a very wonderful step of by using the media, by advertising through relevant advertisements, even on phone. Our ringtones have been hijacked in a way, but still it is for the good of others, for all of us. So next, there are certain do's and don'ts, and better follow these do's and don'ts all the time. Basically, don't get close to anyone. This is what is a don't, and don't touch your nose or eyes when they are especially wet. So, and don't spit in the public. So these are.
Mr. Vijaykumar, please don't disturb. Kavi Karunya.
Should I share it again?
Yes, sir. Yes, sir.
Should I share it again?
Yes, sir. Yes, sir. They disturbed.
Okay.
Please, participants, please cooperate with us.
So our government, our app, our government websites are also very active in promoting all the safety and preventive hygiene right now. So we can get all this information there. I'm just trying to collate them and give a proper presentation to them. So in this immediate preventive measures, what are the guidelines of WHO? So personal protective equipment is one of the important aspects to avoid any social contacts or the different preventive measures I have talked about earlier. So under this preventive personal protective equipment, we have gloves, medical masks, goggles, and face shields and gowns, as well as respirators and some certain procedures. So these are the very important things. You can see there is a huge surge in the requirement of PPEs now, and many innovations are happening in this PPE area of current research and development, ventilators and all those things. So it has taken India. It is a very proud moment in a way, I should say. Based on the exigency or emergency of the situation, ICMR has taken recommended 2 RT-PCR kits for coronavirus without this validation by any FDA or Europe or something like that. So we are taking very drastic steps to contain this particular, because we are a 1.3 billion community citizens. We have to take some tough measures. In that scenario, very strong decisions are being taken. As you can see here, ICMR recommends 2 RT-PCR kits for coronavirus testing, very fast testing. Now coming to the how the data, how the infectivity is across the country. As you can see here, after the lockdown, there is a drastic decrease, only up to the infectivity or the infection rate has decreased to around say 12% or less when it is across the country. Okay, the growth statistics of the country, and you can see here one of the best is the Kerala and Karnataka government. They have taken very strong measures. From the lockdown point of view, Andhra seems to have performed very well. So this particular thing also gives good, but the poor performers are Gujarat and Maharashtra. They are unable to properly contain the whole by lockdown, or means there are many resurgence or demonstrations, protests going on, and that is one concern for that from a national point of view. So what is that we can learn from till now is lockdown and social distancing, the most effective steps in containing COVID-19 pandemic till now in the country. So this is one thing. So next we will go to the available patient data. Let us see how the patient data is across the world. And you can see here Europe and Europe, especially Italy, Spain, and America have contracted or most infected states, most infected countries in the world. Then we have the confirmed, and you can see that China here. I'm not able to. Okay, sorry. So China is the one which stabilized the fastest. Once it reached around 85,000 confirmed cases. It reached, and it is very surprising and very intriguing that out of 85,000, 80,000 have already recovered. I think around 75 to 80,000 already recovered, and but the recovery is because of individuals taking hygiene conditions. This is also one good news. So you can see here more than 16 lakhs of confirmed cases across the world and 99,000 deaths. That is huge number, and still it is increasing. And this is by case comparison, where Southeast Asia is 14,000 alone, as the green color is showing you very less amount of infectivity. Europe is maximum, then America, and then we have a small percentage of Western and Eastern Mediterranean, and Africa is the least. So most highest cases, as I told you, United States, Spain, Italy, and Germany. So we can get back to these things again if we need. So from an India perspective, this particular website gives very updated information, and right now we have already reached, this is 2 days back information, and right now we have reached around 12,000 total active cases, and our deaths, things are also increasing. As I told you earlier that we will talk about this again when it comes to the India demographic thing. Here is the patient gender differences. Awaiting the details is still, we are awaiting the details of the gender up to say 75 to 80%. In that also only 60% to 70% is male, and the remaining 30% seems to be female infected patients. And as you can see from the nationality point of view, 90%, 95% seems to be Indians, and the remaining we are still waiting the nationality of the patients. I told you earlier that age is a very important factor, playing a very important role here. The number of deaths that have happened are around 31 to 50. So this is another important aspect here. So, but as far as the news goes, old people are most affected, and the death rate is very high in the old age, and this is especially because of additional complications which are coming up. So India has divided the whole country into green zone, orange zone, and red zones, and we are trying from, as you all know, that from May 23rd onwards, the green zones will be opened, and the red zones will be more carefully monitored for next steps. So all this data we are okay. This is AP because my concern is with respect to AP and TN. Guntur and Vijayawada are most affected, and next is Kurnool. So we are still trying to come up with a strategy to contain COVID in AP and AP especially. So these are the district level details. But all these things set aside, what is that we should do with this data? What is there any importance to this particular data? So what we try to do is there anything going on about modeling aspect? That means, can we predict the future and what is going to happen in the next 3 months to 1 year? So that's what. So we have around 5-6 groups in the world which are working on taking this data and trying to model. So my current assignment is also to try to model and come up with a better model than the existing. So I'm trying to do that. So first one is JHU, that is a Princeton University model, which has predicted very exaggeratively. They predicted that we should get around. So these are the 7 modeling groups which are working in the modeling the spread and infection rates of COVID-19. So just now I got one news that science has published from Harvard. So they are trying to predict, as you can see from the graph, the bottommost, the second and this one, that in winter, that is after summer, and it has been scientifically proved or scientifically interpreted that temperature and humidity are slowing down the rate of infectivity of COVID-19. So that is a good news for India, but we should not take it lightly, because the next coming after summer, that is rainy season and winter season, may be a real pandemic and real tragedy, or if we don't care, if we don't follow the restrictions as we are following right now. So we have to be very, very careful, because that particular environment will be extremely conducive for the coronavirus. So similarly, these people have proposed, or without interventions, that is public intervention, that is government interventions, like telling the governments to take these steps and those steps. Similar interventions we could have. They predicted that 30 to 40 crore infections were predicted, and by next 2 months, by May, they predicted 20 crore infections. But India has done a very great job in slowing down the overall infection by taking lockdown and other interventions. So that is one good news. So there are 3 models generally we are following. That is SIR model, susceptibility, infectivity, and recovery. And the SCIR is susceptibility, exposure, and infectivity and recovery. So these are the 2 models which across the world people are using for epidemiological studies. So in this case, another group, path group, has predicted certain things. So let us not go into much of the detail. And one from Ohio State University. Sir, nenu oka talk lo unna, online talk lo. So I'm sorry, yeah. So most of the pandemics follow exponential curve, and we are here trying to get the flattened curve. We are trying to flatten the curve, and the epidemiological models are here. So India will enter equilibrium. This has been more realistic model here present here. That is epidemic size of approximately 30,000 by May 2020, and almost we are reaching that particular prediction. So RO is number of infections through one contact. So it is 0.43. It is much smaller than the rest of the world. That is a good news for us. And the current model does not account for factors such as weather and humidity. So this is where we are trying to model using the existing data. And the Cambridge model predicted that lockdowns can influence the bring down or flatten the curves of infectivity, but 21 days lockdown is not effective. So then they have predicted that lockdown, continuous sequential or in a temporal lockdown may be working out, but 49 days is a good strategy to have complete or almost complete reduction in the infectivity. So this is another model, and similarly we have Imperial team's model as well. I am working on that. That is the current affairs that is going on. So let us come to the R&D effect in treatment and prevention of this. So I have another few slides. I will wind it up fastly. Okay, so this is a summary slide which gives you, as earlier speaker was telling, to complement his answer, there are total around 41 diagnostic technologies which are being studied and being approved at the highest level, which will come into the market soon. And another treatment of 23 are treatments, okay, are being studied, and that is drug trials are being conducted, and vaccines, 5 vaccine method, I mean 5 5 different vaccine technologies are being developed and being considered at the FDA level. So this is what is the simple summary slide of all the things in a very clear. Okay. So I can share these data if you want. Right now, let me move forward. So there are many drugs which are being used also. So the most important target candidates for drug targets are 3CL-Pro, which is a protease for the proteolysis of viral polyprotein, and the drug candidate is lopinavir, and PL-Pro, that is papain-like protease, that is another, it is also being, and RDRP, this is another enzyme. Okay, so these are the important drug targets. As you can see, S protein is there, transmembrane protease is there, angiotensin, this converting enzyme 2 receptor is there, and 82 receptor is also there in this. And their respective drug candidates which are being under consideration and being taken to the next phase trials. So the other important thing is that just now, only today, we got this news that Telangana moved to get nod for plasma therapy clinical trials. And plasma therapy is, as I told you, that a person who got infected and he suffered the symptoms, and after that, once he recovered from the symptoms, he is expected to have generated certain antibodies. So this is what we are trying to cash in, where the blood of that particular person is taken and the antibodies are purified or separated, extracted, and these particular antibodies will be again given to the person with the severe complications or severe infection in the COVID patient. So this has shown some promise. So already Kerala has implemented, given a nod for this. Now Telangana also taken its steps towards that. So a blood bank is being developed, which will take these blood samples and process for the plasma and antibodies, and will be trying to do. I think this is one of the fastest way of reaching out for treating this particular COVID infection. And next will be that on the vaccine development, and later will be drug development. So this is the phased way we have to do, but immediate. And as I told you, I have been working on the vaccine development. We published one preprint right now. We are considering it for publication, and we have done for most of the structural proteins, and we are trying for a project on this particular thing. So it has a huge impact on the economics. So I don't want to go into the details right now. And many efforts are being taken by various people across the country. Many innovative ideas have come. This is herbal medicine, using herbal medicine, how to contain coronavirus. This is the next level, that is animal testing phase is a very difficult and extremely critical step for coronavirus, this one. Okay, let us now have, so this was the committee sitting yesterday, day before yesterday. I would like to conclude one simple thing, that prepare for the upcoming rainy and winter season, which shall prove more favorable for coronavirus. And as you can see, temperature and humidity slowed down the growth, and hence infection rates of SARS. It has been published and predicted also. So let us be very careful with that. And these PPEs, you can see here, 89 million masks, 30 million gowns, 1.5 million goggles, and 76 million gloves, 2.9 million liters hand sanitizers have been used and needed under this PPE. So huge effort is there. Medical and biomedical, especially medical and biomedical infrastructure or equipment are more in need to contain this particular COVID as such. And people are coming up with multi ventilators, one ventilator which can support simultaneously multiple patients. So this is one video we have got, and Mahendra, we should thank Mahendra for developing a low-cost ventilator with essentials. And I think that's what I will say. So AI and ML, we have predicted vaccine candidates. So now what we are trying to do is like it is shown here. TCS is using for drug discovery. We are using for vaccine candidate prediction basically. So with that, I should. It should not be like Ministry of Ayush has recommended, and India being one of the oldest civilization, we have had and we still have many home remedies. So let us go back to our roots and follow the cleanliness which we were asked to do in the past. That is the best solution right now to avoid any type of COVID-like pandemic to affect us anymore. So Ayush recommendations are given in this slide, like drink warm water throughout the day. And the same thing Chinese people in Wuhan have done, and they control the whole thing. And the daily practice of yogasana and pranayama, spices like haldi, that is turmeric, and dhania and garlic are highly recommended. And Chavanapras, immunity, basically immune boosters with golden milk, that is haldi, that is turmeric powder, and milk is again recommended. Nasal application and oil pulling is another one. Oil pulling removes all the bacteria and all infections which are in your mouth. So that is one of the best ways of avoiding any infection during dry cough, and lavang has to be used. So based on this, so I will stop here at this particular point, and I will be happy to take the questions for now.
Hello.
Hello.
Sir, very nice presentation, sir. This is Dr. Gopi from Cardamom Planter Association College, Bodin Aikanur.
Yes, sir.
Actually, the presentation was elaborated and full-fledged information. My doubt only is, nowadays people are going for plasma therapy. That means the taking of antibodies from recovered people and giving to the highly healed people. So what is the rate of rectifying or treating the plasma therapy for infected people? Please clarify, sir.
Yeah. First point is, I told you the nod has been given for clinical trials. Okay, so the results are awaited. But I will also complicate this question by telling that plasma therapy also has another downside, which is called antibody pathogenesis. Okay. So we have to be careful before doing this, but that is the best route right now available. Let us wait and see how the results will be.
Okay. So it's a time being.
I cannot give a concrete answer for this because we are yet to get the data on these things.
Okay. We're still under trial and error.
Exactly. But looks like it is worthy.
Worthy. Yeah. At present, it is worthy.
Yes.
Yeah. Thank you. Thank you very much, sir.
Yeah. Thank you.
Sir, good afternoon, sir. Hello, sir. Good afternoon.
Yes,
Hello,
please
sir.
loudly, please.
Yeah, sir.
Make it a little loud.
Oh, yes, sir. I am good afternoon, sir. I am Dr. Mahendra from Karaikudi College, Sivagasi, sir.
Yes.
Thank you for the presentation, sharing your knowledge to us, sir. I have one question, sir. Is there a chance that person who has recovered from the coronavirus will get a coronary infection again, sir? Sir, please listen.
Yeah. I think he will not get, because we might have developed the antibodies for it, and by the principle of antibodies we may not get, but don't know. I cannot. My expertise is not that.
So. Okay. Thank you, sir.
Sir, good afternoon. Very good presentation. Thank you very much, sir, for sharing your valuable knowledge and years of experience.
Yes, sir. Thank you very much.
Thank you, sir.
I could have done better, but because of lack of time.
Yes, sir. We understand the limitations, sir.
Yes.
Thank you so much, sir. Thank you.
Yeah, thank you.
Hello. Thank you very much for your nice presentation, sir. Your current research reports and how to prevent and how to diagnose, and your recent updates up to that World Health Organization reports. It is highly impressive, sir. Thank you very much for your nice presentation on this science forum. Thank you, sir.
Selvan sir, can I ask a question?
Yes.
Please go ahead.
Professor, good afternoon. This is Dr. Rajesh. Actually, I would like to ask you regarding that incubation period. You said that it is about 5 point something days, isn't it?
5.2 days. Yeah.
Yeah, but somewhere in WHO, in the past, I have seen that it is 14 days of incubation period. Even in news channels also, it is not so clear. What I would like to ask you is, actually, within that 14 days, would we develop any symptoms? Or once after infection, after that 14 days of incubation period only we get infection? Because some people say that it is by 5th or 6th day we started giving infections, and once we get the symptoms, then only we used to spread it. So in that case, I would like to know whether we show whether the symptoms may appear within that 14 days or after 14 days of infection.
Okay, so whenever we are talking about patient data and interpretation from patient data, we should be very, very conservative. That means we should have a very broad range of, because I told you, it is the age and also the immune response of the body, right? So on average, even WHO, I also read the contradictory stories on this. Okay. But 5.2 days is the most commonly said incubation time, for after which you are going to see the symptoms. 40 days, I don't think, but 40 days is not, I think it is not the correct number. I think I have read from 5.2 days to 14 days is the incubation period before you get to see the symptoms. That's why it is getting complicated for us whether the person whom you are testing is positive or not positive, because we don't know exactly from when he got the contact and how many days it has been incubating in the body.
Okay, thank you, sir. And another question. Actually, when we talk about that antibody treatment, just now you spoke about, that giving antibodies, collecting antibodies from the required patients and giving it to symptomatic patients. Yes, so in that case, actually, when we give the antibody, even the antibody being a protein, our body would take it as a foreign protein, and even it acts against that foreign protein also, isn't it?
Very true. Yes.
Then in that case, how it would help that person to recover from the COVID-2? Even if the antibody is taken as such, and if it is been assimilated as a own protein by the body, then only it can use it against that coronavirus. Otherwise, what happened? That antibody will also be taken as a foreign protein like a coronavirus. Then the body try to produce antibodies against that antibody what we given from outside.
As I told you in one of the other questions, that there are complications, and also there is something called antibody based pathogenesis. So that's what you are actually talking about. We have to wait till such a thing actually comes out, and we need to take care of such things, and that is the purpose of these clinical trials, not for clinical trials. So you are absolutely correct. I agree with you.
Thank you, sir. Thank you very much, sir. Thank you.
Sir, I think it is time to conclude our session. With the permission from Professor Selvankumar, I take the liberty to thank Professor Prasad for giving you this wonderful lecture. Thank you so much, sir.
Thank you very much. Okay.
So, is Dr. Naveen is there? Dr. Naveen? So we have Dr. Naveen here.
Hello, Naveen is there? Okay, we proceed the Mahima, then we will contact.
I think she's having some network issues, sir.
Okay. Wait.
Sir, you please extend the session for a technical discussion. I will now talk with Dr. Gomadi and see whether he
If Naveen and Mahima is not there, you can call the next speaker.
Yes, sir. Yes, sir. That's what I am
asking. We can reschedule their presentation.
So, Sir Selvan, sir, until now, you please handle as a discussion session, sir. I will.
Yeah. I will connect. Please wait.
You find out with the other speaker whether they are ready.
Sir, Selvan, sir, you please take it as a discussion session, sir. If there are
Okay. Please proceed. I will communicate with Mahima or Naveen.
Yeah. You can call and find out whether they are ready.
Mahima is online. Hello, Mahima.
Hello, sir. I am in the line.
Yeah.
Hello, sir.
Yeah, you can call her.
Sir, can you hear me?
Yeah, hearing.
Yeah, probably you can invite her to speak.
Hello. Yeah, let's invite.
Yeah, please initiate.
Yeah, now I introduce Ms. Mahima. He is an assistant professor, Department of Microbiology, Muthaimal College of Arts and Science, Rasipuram. Now she is going to deliver his topic in prevention of COVID-19. Let's proceed, ma. Hello. What happened?
Sir, Dr. Gomadi is joining, sir.
Yeah, okay. If she's ready, you can introduce.
She is coming in 2-3 minutes.
Okay.
I think we have to request all the participants not to press the present screen button again and again. It's disturbing the speakers.
I think Dr. JP, Mahima is there.
She is having some network issues, sir. That is the
Now
problem.
I could see her. To ask her whether she is ready. No, ask her to on the microphone because it is switched off. To tell her to on the microphone. I am seeing in her screen.
Hello.
Yeah.
She's
Can you
speaking
share the slide or shall we share?
Please share because I'm on phone.
Yeah.
Sir, I have switched on.
Yes, Mahima. Who is? Are you ready?
Gomadi. Yeah, now she's Gomadi.
Mahima.
Gomadi is speaking now.
Okay. Now, sir, Mahima.
Sir, Mahima is in the line.
Are you ready?
Okay, let Mahima go ahead.
Okay, sir. Actually, I connected net now. Hello, sir.
Okay, no issue.
Sir, I am ready, sir.
Yeah, okay. Please proceed. Mahima? Yeah, please proceed.
Good morning to one and all present. Good morning to one and all present. Good afternoon to one and all present here. I am Dr. Mahima, Principal Investigator, Science and Technology Project, TNACST, Assistant Professor of Microbiology, Muthaimal College of Arts and Science, Rasipuram. So I am going to talk about prevention of COVID-19. Next slide. Next slide.
Yeah, madam. Right.
Next slide, please. Next slide, please. Actually, coronaviruses are the family of RNA viruses, medium-sized. It is enveloping a positive strand. The viruses which causes the infection to humans as well as animals. The name of corona, which originated from crown-like appearance of virus. There are 4 classifications of coronaviruses are available. They are alpha, beta, gamma, and delta. Human coronaviruses are causes of respiratory and gastrointestinal tract infections, and which causes gastrointestinal symptoms are diarrhea. Sir, next slide.
Yeah, you proceed.
Sir, next slide.
Yeah, it's a display.
Next slide, please. Okay, so next slide, please.
Yeah, but who is
So
on?
human coronaviruses, which causes infection to humans as well as animals, which causes respiratory tract infections and the gastrointestinal tract infections. Gastrointestinal symptoms including diarrhea. When mutation occur, coronaviruses can be significant cause of human epidemic. Next.
She having some network issues.
Sir, if Dr. Gomadi is here, we can proceed with Dr. Gomadi and we can invite Dr. Mahima last
Yes.
after the session.
We can postpone to Mahima. Please ask Gomadi ma'am to present, please. I will call Naveen.
Yes, sir.
Okay.
Dr. Gomadi.
Okay, can you see me now?
Yeah, please wait. I will introduce.
Yeah.
Dr. Gomadi Aranan is a doctorate from Department of Biological Science, Pondicherry University, 2001. She has worked as a research associate in the CS in Botany during 2002 and 7. Later, she joined in the Volvo Biotech Private Limited and worked there from 2007 to 2018 as a senior scientist, where she had worked on drug discovery and expanded to nutraceuticals and flavor and fragrances. She had developed a replicant model assay system for alpha-like viruses. More importantly, she is having one granted patent from her discoveries, and she has applied 5 more patents as well. So she is now doing her research independently, and she will be presenting her talk now. Welcome.
Thank you, JP. Thanks for the nice words. I started my career with plant biology, and when I joined Evolve group of companies, I started working with drug discovery, and initially I was associated with the DTRA funded project of finding antiviral drugs with replication machinery. So after that, I haven't worked with the viruses. Then we started working on flavor and fragrances and nutraceutical molecules with the synthetic biology, as JP said. Again, my interest started after seeing this coronavirus outbreak and started reading papers. So I thought I'll share some of the papers which I read and share my views with this group so that I can get more inputs so I can start my further work. Now I'm working as an independent researcher. JP, can you project the slides? Thank you, JP. As our previous speakers has mentioned lots of information on how the viruses are originated and how it was spreaded from China and what are all the sources might be. Here I'm just focusing only on aspects of why replication machinery has to be also considered, because lots of research is going on in other areas. Can you just other targets? Can you just change the slide? Next slide, please. Second slide, JP.
The slide has been changed. You please go on.
Oh, okay. So I could not see. Okay. The this virus, actually, this coronavirus, SARS-CoV-2 or SARS-CoV-19, as it is named, is a positive sense single-stranded RNA virus. I'll just take my slide. I could not see the slide. Okay. Yes, okay. What is the special about this? Why I have to mention, as many people has mentioned already, because it's a genetic material of positive strand single strand RNA can function as a genome as such, and as well as a messenger RNA. So as instead of a virion particle, the genome itself can infect humans, as well as, though the infection rate is very low, but it can infect as such, because it need not depend on for the first infection, it need not depend on first proliferation, and need not depend on the RNA dependent RNA polymerases. That's the reason, because the positive strand acts as a genome material for the replication. And the first proteins produced or auto-associates with the membrane, and it forms vesicle, and they are the one which they started the replication factors and links with the successive replication steps, like performing subgenomic RNAs and making into capsids and other things, packaging all things. It protects the replication intermediates also. The first proteins protects the replication intermediates from host defense responses, so that they can escape the host defense responses and they can keep on multiplying. And the genome of most of the positive strand RNA viruses are very small. It's very small. Like, for example, the BME virus has only 3 ORFs. But the coronavirus is the largest known RNA viruses. It has got about 27 to 32 kb. The present COVID-2 virus is having about 30 kb size. Now, coming to the SARS-CoV-2 genome replication. Actually, now it has got a replicase transcriptome complex that is actually coded into ORF1 and ORF1B. So both of them jointly together, you can see the picture which is there. Both of them jointly together called as the ORF1AB. They translate into protein, and it forms a very large protein, and it is actually comprises of 15 non-structural proteins. All the other ORFs are coding for the structural proteins. The NSP1 to NSP16 proteins are cleaved after the long polypeptide is formed. The cleavage, everything has been done within this by the proteins which are coded by 1AB. Why we should target the replication machinery? Actually, the main focus in the recent days in the papers and all targeting the receptors and uh, the immunomodulators of human. All the things are going on in a very detailed manner. There are many studies are going on. I felt there is actually a small maybe the results this researchers may come little later, but there is a small lag or there is a small negligence on targeting and the viral replication machinery. Why we should target on the viral replication machinery is that actually it is vital and it is a survival mechanism, and that is the key for my main multiplication. After the virus enters into the cell, it hijacks the human proteins and it started multiplying. If we can target the genome multiplication itself, the virus can be harvested completely and it cannot be propagated more. So that is one of the major targets can be taken. And one more thing is that we have seen in the previous presenters, Dr. Madhivanan and Dr. Pillai, all of them, they have in detail they have explained that this virus has an origin and it has got very close relationship with SARS COVID. The SARS-CoV2 has got very close relationship with the bat SARS-like genome. But when the one of the studies which was published in Cell and Host Microbe, it shows that actually there are lots of when they translated the sequence into amino acids and proteins, they found out that there are lots of actually mutations happened. So these mutations has caused and maybe these mutations are the reasons that this coronavirus is more severe and spreading faster, and it is very difficult to tackle also at this point of time. Actually, they identified actually 380 amino acid substitutions when compared to the previous SARS and MERS CoVs. But out of all this, actually there are a few proteins which are not at all altered, or that is 7A, which is actually and nucleocapsid genes, and the envelope and matrix accessory proteins, they are all not at all affected or not at all changed. And but the one of the major alterations has happened in the in substitute happened in the NSP3, that is non-structural protein 3 and non-structural protein 2. They are the initial proteins which are formed in the multiplication or replication machinery. So why these changes have happened, or these changes in the amino acids may be one of the major reasons for the virulence factors of the virus. So targeting this may be helpful for finding out or repurposing the existing drugs which can effectively control this protein. And this actually, and here also we have actually RNA dependent RNA polymerase, and there are very few substitutions in this. And the non-affected transcriptional protein is that NSP13, which is the major target, and it is actually binding with the now we have seen that there is already drug available for that. Can you change the slide, JP? Third slide. Sorry, fourth slide. Fourth slide. Hello.
Akka, I'm sharing your screens only. What number of slide you want to show now?
Now 4.
4, huh? 4 is replication machinery.
Yeah.
Right.
Okay. Sorry, now 5. Can you just move to 5?
Okay, now 5 is on.
No. What are all the targets already, which is actually they're common in all the other SARS viruses they have already screened. That is one of the targets can be PL-Pro protein, that is a papain-like protease protein. Why it is very important is that it cleaves the polypeptide formed by this the genes 1A and 1B, because it forms a very large, and it is it's having 16 genes. All the 16 genes are cleaved only by this proteases. So if you can target this this region, it will be very it will be very crucial that the virus cannot multiply, and moreover there is no FDA approved drugs in this region for any of the SARS viruses also. So if you can find a drug which can affect, which can target this, we can the viral replication can be completely controlled, and as well as the TNP5 protein, that's also sorry maturation. It works in the maturation step. So if viruses cannot mature, and helicases can also be targeted, because it is indispensable, and there are actually few inhibitors already in trials, that is DNA optimers, but there are actually side effects which are reported for this drug also. Can you just go to the next slide, that's slide number 6. Then, so the same slide is continuing the targets for the viral replication. There is NSP12, which is actually which is also a conserved protein, and that is the RNA-dependent RNA polymerase. As we already just mentioned that this is actually crucial part, because this is the one which is actually taking the transcripts and genomic RNAs and multiplying into many copies. So actually the already available drug that remdesivir is that is actually in the clinical trials. That drug is already administered for some of the patients in France. I believe they found there is a cure, but it's not completely. It cannot be completely attributed whether it's because of this drug, but there is a cure cause. But problem is that when of the patients which are administered, they face lots of toxicity. Though actually this target is supposed to be non-toxic, because this target is not related with the human proteins, but somehow this way may be reacting with some other targets of the human body, so that is causing already lots of side effects. So again this target, and there are lots of computational biology which are available that very hello. Okay. There is a lots of noise. I don't know what is that. Okay. And so most of the studies papers which are published, they all use the computational methods or modeling methods to find out whether the targets are lots of targets, like even proteases and receptor proteins, everything they have designed, and they have by computation method they have screened with drug databases like Zinc and other places. And some of the preprints are some studies which are going on. They also screen some flavor molecules also. They found that even camphor, patchoulol, or this all this, they are actually are actually binding with these sites. But whatever the binding, but the recent paper published in Science, that is the one which is actually crystallize the protein, that RNA dependent RNA polymerase, and they have made whether that remdesivir is actually binding with that, and whether the proteins which are in the key places are all receiving that or not. I have presented this in the picture, which is just published recently in Science 5 days ago. More studies are yet to come with tested on animal models or tested on in vitro cloned in cell or human cell based assays, or some other assays like how we did in our DTRA project. We cloned the entire new replication machinery in the yeast, because yeast is also it can express, because it's very all the receptors and other things which are there present in the plants or in the animals are also present in the yeast to receive the receiving viral genome and replication machinery. The host pathogen interaction, the virus and the host is more or less similar. So it can be cloned in the yeast, so that yeast clone results we can produce the results faster, and the stability is also very high, because the maintaining cell lines and the passaging time, all these things and the replication time is very low. And moreover, yeast is also having its own report systems and all. So I haven't seen any. If some other people has shown, please share the results. And no in vitro tested results or no animal model tested results for even the repurposed drugs are done. So we have to start with those, with maybe a consortium of people or something like that. So that's what is the last slide is about. So at present, yes, there is a dire need that we have to immediately find drugs with whichever available molecules, because repurposing is easier. But still, the hydroxychloroquine or the remdesivir or all, they are also having lots of side effects. How many days you can administer with these drugs with the side effects is a big question. So, but since we have to save the patients, yes, fine. But the SARS epidemic has started long ago, and swine flu has started long ago. That there is recurrent occurrences of these viruses again and again with maybe slight modulations. As already told that there is changes. These viruses can undergo mutations, maybe slow or faster, and also they can undergo recombinations with already if there is the person is carrying already other viral infection. And there are reports that even the positive single strand virus also undergone lots of crossover with other positive strand viruses, which are like common cold, common flu, or all the things. So we can expect lots of new outbreaks maybe coming, or it should not come. If comes, we should be prepared to face this. So definitely new novel compounds with less toxicity is also a parallelly important task to be carried out. So assay designs with maybe with this new genome sequence has to be started, and because the replication machinery is the key and pivotal plays a pivotal role. And as well as in this present genome, there are actually lots of the changes has happened with the NSP2 and NSP3 proteins, which are the early proteins. So those has to be targeted. That's my conclusion. Thanks for the opportunity. Really, I appreciate the organizers at this organizing such a great event and bringing definitely very strong young minds to the experienced researchers in this platform to present. And thank everyone. Thank you.
So thank you very much, Dr. Gomudi. Thank you very much for your vivid presentation. I request the audience to restrict 1 or 2 questions only. We are running short of time. We can have maximum 2 questions quickly, please, if there are anything.
Hello, ma'am.
Yes.
Hello.
Yes.
Ma'am, what do you think about the administration of monoclonal antibodies?
Already Pillai told that about this. Yes.
Yes, ma'am.
We can. Actually, I'm not expert in this field, so I cannot Hemant, yes, at this hour, we can do whatever possible ways to save the patients. And but there should be like a clinical trial or precaution to be taken, because already one person has asked what will happen to autoimmune patients if there is a viral infection. What are the treatments? Those complications are there, and as virus enters also, the immunity of the person also alters. So there should be care taken. That's all I can say.
Okay, ma'am. Thank you, ma'am. Thank you so much.
Welcome. Any questions?
Sir, can I have a question, please? Hello.
Yes.
Yeah, please, sir. One last question, sir.
Madam, this is Dr. Rajesh again. Actually, while going through your presentation, I got an idea. Maybe it's very crude or layman, but while listening to your presentation, I have seen that there are a few amino acids which involve in the preparation of their structural protein and all, even in the replication also. So my idea is, my just crude idea, when we deplete any amino acid which is non-essential to our body, when we deplete it for some time, can we prevent that replication? Because as you are working on that amino acids and all, as you well aware about the information, if you think in that way, maybe is there any one or two amino acid which we don't need essential, which is not at all essential, maybe that we deplete and we can stop the protein synthesis. By means of that, we can prevent the spread of virus.
Yeah, actually, I got your question. So, yeah, you mean that the receptors in human
If
body,
possible, please
in that,
open your video.
I'm on video only.
No, not for you, ma'am. And next, I am asking.
Okay. Actually, yeah, but gene editing in human body is not that easy at this point of time. Maybe the like CRISPR-Cas child has been tested, born like that. If in the future we can think about it, but what are the implications it will happen? All the things. It's a long way to go. Definitely, it's a good idea.
Thank you, madam. Thank you very much.
Welcome.
So I think, Sir Salvan, sir, shall we move on to speaker, sir?
Yeah. You proceed.
Yeah, sir.
Introduce Naveen.
Yeah, I'm taking the privilege of introducing Dr. Naveen. He has completed his master's degree in Gulbarga University during 2005, and later he worked for a few years in the R&D unit of Merck Millipore, India, and he got a fellowship from Taiwan government to pursue his PhD at Academia Sinica, and completed his PhD in 2016. So he has postdoctoral experience at Taiwan and as well as Australia, and presently he is doing full-time research. He is a full-time research fellow at Biomedicine Discovery Institute of Monash University, Australia. He is concentrating on the structural and functional studies on virus and bacterial proteins involved in infection and challenging human immunity. More importantly, Dr. Naveen is currently working on unraveling the structural mechanics of novel coronavirus COVID-19 and human targets. So with this brief introduction and on behalf of the organizing committee, I welcome Dr. Naveen to give his lecture. Please, sir. Dr. Naveen, you can please continue the session.
Hello, Naveen. Hello.
Dr. Naveen, you have to join the session and then unmute yourself. Dr. Naveen, you have to click on the Chrome close to the address and you have to allow the microphone in the Chrome itself, in the same page. Just the end of the address bar, there is a into mark on the phone icon in your address bar. In your address bar, web button. Meet.google.com is there. So that is a web address bar. So at the end of that, there you have a button. Not there. Or you can go to settings. Yeah. Yeah, you have to permit the microphone. Can you refresh? Can you refresh the page? Yeah. Now you. Oh, okay. No, don't cancel it. Open system preference, and you have to enable the microphone. Yeah, you have to. Yeah, correct. Yeah, you can quit and restart. No problem. You have to quit and restart. Yeah, close and refresh the page. That's all. Yeah, now refresh the page, the Google Meet. I think you have to close completely Chrome and then restart it. Yeah, just the Chrome, you completely close it. Now you have to close Chrome completely. You will get disconnected, no issues. You have to close the Chrome completely and then open it. Then only it will work.
Yeah, Mahima. Hello, Mahima. Are you free? Hello. Shall we call Mahima?
Sir,
Yeah. Okay. Shall we? Hello.
Yes,
Okay.
sir.
Yeah, you can start.
Okay, sir.
இப்ப நீங்க க்ளோஸ் பண்ணீங்க. ஜெயபிரகாஷ்
சார் சொல்லுங்க சார்.
நான் ஷேர் பண்றியா நான் பண்ணவா நீ பண்றீங்களா?
இல்ல நீங்களே பண்ணிடுங்க சார்.
ஓகே.
ஓகே சார்.
Mahima. It is missing. Yeah, you start. Oh, hello. ஜெயபிரகாஷ்
சார் சொல்லுங்க சார்.
இப்ப நீங்க ஷேர் பண்ணுங்க. இதுல பவர் பாயிண்ட் மிஸ் ஆயிருக்கு.
அப்படியே சார்.
ஆமா
பண்றேன் சார்.
What is the problem?
Madam, you can continue, madam. The slide is on.
Sir? Hello, sir? Sir?
You proceed.
Sir?
Hello.
Hello, sir?
Hello. You can proceed. Jayprakash.
Hello, sir?
Yes, you start, ma.
Are you able to see the slides, sir?
Hello.
சார் அவங்ககிட்ட ஒன்றும் ரெஸ்பான்ஸ் இல்லை போல் இருக்கு.
மகிமா. Hello, Naveen. Hello. Hello, Mahima. Or Naveen, are you free? Mahima. Hello.
Hello? Hello? Sir, is there any changes in the schedule?
Dr. Jayaprakash, I think Niyaz Ahmed is online. Probably you can ask him to speak. Dr. Selvankumar?
Yes, sir.
Yeah, Niyaz is online. Probably you can ask him to speak.
Yeah. Shall you start, Niyaz?
Niyaz, are you ready?
Yeah, I'm ready.
Yeah.
So then you can share your slides, please.
I already given to
Jayaprakash.
Hello?
Sir.
JP, sir?
Jayaprakash,
Can you play my slide?
I think network issue on Jayaprakash. Jayaprakash, you off your video, then you connect your mic. It will be better. Dear participants, please.
Sir, all of you are seeing the.
Can you hear me?
Hello, sir.
Yes.
Sir, are you able to see the screen, sir?
Yes, is here.
Yeah.
So on behalf of the organizing committee, I take the pleasure of introducing Dr. Niyaz Ahmed. He is an assistant professor in the Department of Biochemistry, DST, sponsored Center for Biotechnology at the Sacred Heart College, Autonomous, Tirupattur. He completed his PhD from Bioproducts Laboratory, Central Leather Research Institute, Chennai. He has worked on the characterization and evaluation of biomaterials based on fibrin, chitosan, and regenerated cellulose. He has done a postdoctoral fellowship at the Center for Nanotechnology at VIT University, Vellore. He has also received a Young Scientist Award in 2012 by Indian Institute of Science, Bangalore, and another one award from Science and Engineering Research Board, DST, Government of India in 2015 also. So he has joined in teaching in 2006, and he is having a teaching experience of more than 10 years in teaching biochemistry. He is a member of various professional bodies like Association of Indian Biologists and so on. So he has handled a total grant of consultancy project to the worth of more than 30 lakhs. So he is a young and budding researcher. With this short introduction, I request Dr. Niyaz Ahmed to continue the session.
Okay, anyone played my slides?
Yeah, sir. You just talk, sir. I will show it, sir.
Okay, fine.
You can stop your video. It will be better for your sharing your voice.
Myself, sir?
Yes.
Yeah.
Yeah.
Hope all of you hearing me.
Yes. Now your first slide.
Yeah.
Yeah. Please proceed.
Yeah. Can I see my slides?
Yeah. Niyaz, please start.
Yeah. So this is Niyaz from Sacred Heart College. My topic is COVID-19 challenges to researchers. So today we are going to know something about the COVID-19. From the morning itself, we discuss in detail about the various aspects and perspective behind the COVID-19, novel coronavirus disease 2019. So in my opinion, I'm going to give a brief introduction about COVID-19. In addition, we are going to discuss in detail about various opportunities and challenges get from this crisis. Each and every negative aspect, we have to take some positivity. In this crisis, we are going to see various opportunities by fighting against that pathogenic disease, COVID-19. In my first slide, I'm going to, before going to my presentation, I will quote the Benjamin Franklin quote. We are going to get best benefit by sharing the knowledge. While we are discussing each other, we are going to share the knowledge. We have to get some good benefit, good information from this present discussion. Next, I can move on to my third slide. Hope audience can see my slides. Dr. Jayaprakash?
Yes, sir, no problem. You can proceed your talk.
Is Daya seeing my slides?
Yeah, we are seeing COVID-19 coronavirus.
Yeah. Let you go with, I can't able to see my slides. Is that okay for you?
Yeah, it is okay.
Okay, fine. Then you can move on to slide 3. Here, in slide 3, we will give some introduction behind the COVID-19.
Jayaprakash.
Yes, sir.
Please keep it in full view. Then only he can easy to visualize and see and deliver the talk.
Whether audience are traveling with me, I can't able to get.
Yeah, that's a problem. Yeah, okay. Full screen. Yes, can you visualize?
Yeah, I'm seeing now. Fine. That's fine for me. Okay, fine. So regarding COVID-19, so coronavirus disease 2019, the COVID-19 is the full form of CO stands for corona, VA stands for virus, then D for disease 2019 is said to be the that virus first identified in the Wuhan city, China. Actually, it is severe acute respiratory syndrome coronavirus 2 is the correct name. Previously, people used to call it as a novel coronavirus before getting a name from the authenticated agency. So here what we are struggling with SARS coronavirus 2, SARS-CoV2 is said to be the third generation of coronavirus. The first generation is the early 21st century MERS-CoV, Middle East Respiratory Syndrome coronavirus. It was the first kind of virus which give high kind of impact in and around the country. The second thing is said to be the SARS-CoV, severe acute respiratory syndrome. Its mutant form which we are struggling now. In occurrence and development of SARS-CoV2 depends on the interaction between the virus and the individual's immune system. The bitter truth is said to be the treatment behind the COVID-19 is highly depends on the person's immune power. So, however, it must be considered that medical intervention can achieve 100% curative effect. So, the immune power of the system plays an important role. Next slide. So regarding the coronavirus, as I told, it highly depends on the immune system of the patient. So if in case a good immune system personality and the another person affected by the coronavirus, he might be face these kind of symptoms. The symptoms leads to various days of immunological disorders. First, he may suffer by fever, fatigue, muscle pain, and a dry cough. In the day 2, he will feel about the difficulty in breathing. The day of 7th, those still with breathing troubles, and he can move on to the hospital. Under his treatment, if the things goes in the same path, he can move on to the acute respiratory distress, which leads to he has to be switched over to the ICU unit, intensive care unit. If the things are going in the right path, if the patient's immune system respond the treatment in the right aspects, the fever ends and the shortness of breath stops. So he may be recovered from the illness and he will be get free in the day of 22. But in the same time, the person's immune system cannot perform in the right path. It is not obey with the treatment protocol given by the physician. He may be go to death during the day 15 to 18th day. So it is about safety that the person immune system plays an important role with respect to the treatment behind the coronavirus symptoms as well as illness. Next slide, please. So regarding the statistical report, I don't want to give much detailed information about the statistical report because Dr. Madhivanan sir from Director of CS Botany give detailed information about the statistical data about the coronavirus affected person and the death ratio. As per my report, it was collected yesterday around 3:45 p.m. report. It said about 19,96,681 persons got affected around the world. It's about positive cases reported. Among these 1,27,590 people got death. So these kind of issue got affected in and around the world. You can see the Indian affected area in the statistical report in the graph of the Asian region. So regarding the statistical detail, SAR gave the detailed information. That's why I not want to give much information. We can move on to next. Next slide, please. Dr. Jayaprakash, sir. Next slide.
Jayaprakash. Hello.
Hello, Dr. Jayaprakash.
There is a network issue.
Oh.
You can go ahead.
Okay, I can.
Okay. Then
So regarding the, as I already told, the immunological system of human, it plays an important role regarding the treatment perspective. A old proverb, we people used to use the very old proverb, it always suitable for all time. Prevention is better than cure. So regarding the prevention, we have to protect ourselves from the infectious disease. It is one of the very good strategy behind the protect ourselves from the disease. If you see the slides, cure perspective is whatever the thing you are take as a treatment, it highly depends on the person immune system. So regarding the corona, how can we protect it? Regarding protection, it's two different strategies. You can protect yourself, you have to protect others from the spreading. So regarding protective strategy, that also well detailed in our previous speakers, my successors, regarding the wash your hands regularly. As per the WHO strategy, they recommended you 20 seconds. Cover your cough and sneeze into the tissue or into the elbow. Use a mask if you have respiratory symptoms or carrying for someone who does. If you have a fever, cough, or difficulty in breathing, seek our medical care early. These are all the compulsory things you have to follow as per the WHO directions. Okay, so next we can move on to the right title of my presentation. What is the what research we have to?
Excuse me, Jayaprakash, please share your slide.
Sir, my network is lost, sir.
Yeah,
So
that's why I understood.
If possible, can you please share the slide, sir? It is there in the group mail.
Okay, I will try. Please wait. You proceed, sir. Yes, please.
Yes, yeah. Okay, fine. So regarding the what research is going on and what we have to do, that is the key role of the researcher. Regarding the RT-PCR based diagnosis kit is only available and acceptable method at present. In India, it costs around 4,500 as per the government norms per test. It needs a nasal swab as sample with 3 to 5 hours of assessment to get the results. Based on the results, you can be able to understand whether a person is affected or not. He may get yes or no results. So, regarding the research perspective, diagnosis of COVID-19 is at present we are following the RT-PCR. It will give documented evidence that a person get affected or not. When we are trying other kind of testing tools, we have to obey the WHO guideline, which also highly documented by my previous speaker, Dr. Prasad Barna from Andhra Pradesh. What are the regulations we have to follow before you designing the rapid test kit for detection of COVID-19? So regarding the present scenario, the immune system plays an important role. Even though, hope you might seen the slides, it will give the clear picture about our immune system responding. A coronavirus get entry into the system, the macrophage, epithelial cell, differential count, various cytokines are like B cell receptor, T cell receptor, cellular immunity, human, all the things it plays its level best. But due to the nature of the coronavirus, sometimes it changes its nature, which leads to various kind of issues. That is a main drawback behind the treatment protocol. When we go in detail about the diagnostic perspective, next slide, please. At present, whatever the available methodologies are available in the regarding the diagnostic methods, all the available methods has to be converted for the COVID detection, the CT image, etc. So these kind of available existing methodologies somewhat made convenient to the detection of coronavirus, COVID-19. So next slide. So regarding the WHO, World Health Organization plays an important role in various perspectives. It plays a major role, gathering latest scientific and finding research in and around the world and comparing the database. They are working day-to-day basis. It also updated in their website. They update on research activities for novel coronaviruses, international clinical trials performing in and around the world, COVID-19 emergency use testing procedures, solidarly clinical trials for COVID-19 treatment. So in around the world, WHO plays an important role. It gives the detailed input about the various issues takes place around the world to the general people as well as the researcher to design their protocols effectively. Next slide. In India, ICMR plays the important role, a vital role also. It conducting various projects, collaborating among other countries for facing these kind of crises, giving guidelines to Government of India. It also declaring the what are the do's and the don'ts in the various times when the severity gets increases due to the COVID-19. In addition, next slide, please. Hope you people are aware about the DRDO, Defense Research and Development Organization. Along with the defense, it plays some important role with the name of DRDO LSRB, Life Science Research Board. While I am doing a postdoctoral studies in VIT University, we did some project in the DRDO LSRB. They are doing various kind of research for the benefit of soldiers and the general public also. At the time, we designed some mosquito repellent for the benefit of soldiers. They're working for the general people also. They give various project call fairs and various kind of projects they are performing for the benefit of general people. Next slide. So DST SERB, Science and Engineering Research Board, working good for the benefit of humanity. Call for various projects and the proposal under the NANA mission as well as COVID-19. At present, we are also writing some project proposal to Science and Engineering Research Board regarding the point of care rapid test kit by using some nano sensors with the help guideline given by the WHO. So regarding these are all the various funding bodies and the research and consultancy agency, including DPT, Department of Biotechnology. It gives lab biosafety. When we are working on those things, we have to take care about the safety measures also, because we are not working with normal pathogen. It is highly pathogenic virus-like factors. We have to take care about the system also. Conducting special series on COVID-19, these kind of various events take care by the Department of Biotechnology. Next slide. So these are all the various funding bodies. They are working and they are ready to collaborate with us, various researchers in their perspective. So as the domains are available, we have to think different ways of thinking. So differently for the betterment of the get solution for the pandemic COVID-19 by using various guidelines given by WHO in world level and the ICMR in India level. So next slide. So what are the key perspective we have to take care? First thing is said to be the fabrication of low cost diagnostic kit, because we don't know what is the right number of positive cases. So if kit is available in the low cost, means it is affordable by the general people. He can able to purchase it and test it. At the same time, whether he can know whether he is positive or negative, then he can move on to the other kind of RT-PCR like confirmation test. So active research on existing and alternative therapy. at the same time, while we are struggling with the existing therapy, we have to take care about the alternative therapy also. In the morning session, Dr. Madhivanan give the detailed information about the various alternative therapies that also keep into the account for the treatment of these kind of issues. We have to design the low-cost PPE, personal protective equipment, for protective clothing, helmets, goggles, or other garments or equipment designed to protect the social workers, because they have to be center of attraction, because they are highly susceptible to this kind of pathogen. So those social worker or a physician who are highly susceptible to this kind of infection, they have to be protected from by using low-cost PPE that will be highly helpful to them, as well as the sponsoring agency. They can able to purchase it and give it to them for the benefit of people. So I come to the end of my presentation. What we have to get various kind of this kind of situation. We have to take into the account that there are various, we are growing, the world gets growing. When we are growing, some kind of negative impacts may come and disturb us. At this scenario, while if you see that slide and then the thank you slide, when we're growing, there are kind of negative impact like that black mark. When we have to mask the black mark by using some new technology, the change into the technology for the benefit of human, so we can mask these kind of issues. So these are the things I want to discuss with you. You can next slide, you can post your questions in the chat box. At the end of my presentation, thank you so much for the opportunity given by Biological Science Forum, and especially Dr. J.P. Jayaprakash sir, Selvankumar sir, Vaseeharan sir. By using this opportunity, I would like to give my special thanks to my PhD scholar, my future Dr. Rahul, for preparing PowerPoint presentation, the various kind of technical discussion held with you. Thank you all. Thanks for the opportunity.
Thank you, Nias. Very nice presentation. You are given a very good idea about the opportunity and the notification given by the Government of India, DST, DBT, etc. It is valuable for all the scientists and faculty friends for applying COVID-associated projects. So thank you for your nice presentation. Next, I invite Naveen, Dr. Naveen.
Yeah, I'm ready, sir.
Yeah.
Can you hear me?
Yeah, share your first slide.
Okay. Hope you guys can see my slides.
Yeah.
Okay, cool.
You can, if it is, if any problem with your network, you can off your video.
Oh, yeah, okay. The network has no problem.
No issue with it. Then proceed.
Okay, sorry.
You share your entire slide. Yeah, okay, good.
Okay.
Okay.
I'm sorry for the disturbance before.
Yes.
I apologize for miscommunication. Well, I'm going to talk about what are the structural and molecular interplays happening. Interplay means how the virus is going to interact with the host protein. That's what I'm going to talk about. That's the main junction where it is happening. As we know that the current pandemic virus has started from Wuhan, or the China, where you say, but it has been spreading all across the world in the matter of time. But their genetic modification is keep on changing. As the spreading is increasing in the time course, it's still kept the same genotype, which is shown in the blue color, but they have the same number of mutations. But as the time goes, the mutations has changed, and that made a new variants of the virus. In the bottom panel, it shows the diversity and intensity of the mutations at the different points. So as it is spreading all across the world, they have going immune passages happening, because a different ethnic background has different rate of an immunogenicity, and as well as the changes, adaptations happens. So that causes some sort of a minor SNP, or single nucleotide polymorphism, or point mutations, we can say. That does changes a lot of thing if and only if it has a mutations in the receptor binding or target binding. So let's talk what are the things are happening in that aspect. So if you look at just a virus binding, like what we have the virus binding to in a receptor, which is a well-known and a first known receptor called ACE2, and it replicates, and this is what everyone has told. But in reality, it doesn't look like this in simple. It looks like this. Is a hell number of viruses, tons, like millions of viruses sit on a single cell. Like if you look at this, is in a single cell and binds with thousands and thousands of virus docks on the virus, on the host. So which means like when the virus is in a very close vicinity with the host cell, it really may not require any sort of a receptor. Because receptor-mediated endocytosis or receptor-mediated entry of the virus is one of the more known potential pathway. But the virus does hijack the human cell or enter the cell with several different pathways. But presence of ACE2 or receptor will enhance. If it is absent, probably it will reduce 20 to 30%. That's why there are n number of ACE2 inhibitors are there. ACE2 inhibitors are dated back to 1974, 1975, and every cardiac and patient and every diabetic patient, they are very commonly using ACE2 inhibitors. And there is then, if that's the case, then why the death toll is still increasing and the infection is still increasing? So possibly there is other targets, and it is also still mediated by several other entry modes, because targeting only one entry is not going to block. Something like you just close the main door doesn't mean the thief cannot enter your home. So in another way, let's look at the virus, and which is going to interact our ACE2, but they does have certain other kind of an activators or an enzymes, which are called as a furin, and another one is called as a TMPRS. So today I'm going to talk about this red box, what is happening at that junction. That's what I'm talking about. And in most of the study, I'm presenting the results from our research and what we have published so far in this regard. The first and foremost thing which we published in last month is the first is complete sequence alignment of an SARS-1 and SARS-2, but that time we are still named as Wuhan virus, but it's okay. So the major thing in the N-terminal, you see an N number of differences, like those differences are highlighted in the red boxes, the mismatches you can say. And one among them is the central one is furin cleavage site. And this is present in the middle of the N-terminal S1 domain and S2 domain. And the S1 domain is involved in the host cell interaction, and S2 domain is involved in the trimerization. When the virus spike protein, as you say, is a trimer, if it cannot trimerize, it cannot infect. It is non-virulent. I mean, I can't say it's completely non-infect, but it's potentially it can not infect. So based on this, we first undertook what are the possible other targets for this virus. So before knowing that, we should understand the overall structure of the spike glycoprotein. But when we published, still there is no structures were available. So this was a hot selling topic at that time. And we published the two modern structures of a spike glycoprotein in a closed conformation, and another one is an open conformation. When the open conformation means it moves one domain from the spike glycoprotein and goes towards the receptor, and where it interacts and hijacks and endocytosis takes place. So this mediates to, in proposing one of the model, like in the different type is at the lower pH or in a closed conformation, the spike glycoprotein, the S1 domain is closed, and eventually it opens and docks the human ACE2 receptor, and then which signals to further enter into the cell called endocytosis. So in that case, if the ACE2 is the only one is the receptor, but we felt that there is another receptor called as CD26, which is also called as a DPP4, and which is also known receptor for MERS. So previous one of the speaker has said probably MERS is not. I cannot say yes or no, but we thought it is yes too, because the residues conservity is very high. And as the ACE2 and the CD26 has shared some of the conserved residues, and which is also involved in the interaction with the spike glycoprotein, there is some sort of potential, but we are not sure how much it extends. So that allowed us to publish in one of the premium journal in emerging microbes and infections from the Nature group to tell that how this spike glycoprotein can interact with another human target like a CD26. And to support this hypothesis or to support this results, a couple of things I would like to emphasize. The two new articles came in the news and reports, like could inhibiting the DPP4 enzyme could threat the coronavirus. Another one is COVID-19 disease, can DPP4 is another inhibited role. And based on our study, and the US company has approached us and take the models and they also made two mouse knockout models for COVID-19 research, that is the ACE2 knockout and DPP4 knockout mouse, so that the mouses are available to test the viral infection so that you can more quickly study, at least in a mouse models. So which further corroborates possibility of the DPP4, because using the ACE2 inhibitors for diabetic and as well as cardiac patients is really a catastrophe. They causes more death for some of the off targets because of the ACE2. Following that, we also need to know, does the spike glycoprotein really appears like this in reality? No, because when it enters the human cell, it undergoes the glycosylation. As the name indicates, spike glycoprotein, which means a glycosylation which will happen. So glycosylation is one of the key to camouflage the virus. So when the glycosylation happens around the spike glycoprotein, it gets immune or it gets hidden from the host antibodies. So antibodies may not going to target the protein part. So in that regard, the glycosylation of the virus will help to hide or camouflage from the host some of the antibodies, not everything. So here I would like to show, like a blue is a coronavirus 1 and green is a coronavirus 2 glycosylation pattern. Although it looks similar in one angle, but if you look at the position wise, the blue is the unique positions and red is the conserved positions, and the unique positions are very well different. So it underlines that some of the antibodies which are being used previously for SARS-1 may not be functional. So, and after that, we looked into what are the possible drug options to block or impede or break the trimerization. As I mentioned, the trimerization is very important for spike protein to functionally active and virus to enter. And fortunately, we saw that an H3N2 is an influenza virus possesses a similar conservity of the residues within a COVID-19. So that makes it very interesting to see whether the drug which is going to involve in the blocking the H3N2, that is the influenza infection, can also block the COVID-19. So there was a first thought, and we just did a molecular dynamics and simulations, and we found the 6 potential target binding sites, and we found one is the most potential target site, that is a C2 position. So, and these spike glycoproteins are showed in the cartoon representation, and let's show you how it looks. Okay, this is the one. This is a drug called Arbidol, and now it is US FDA approved drug, and it is also under the clinical trials for the COVID-19 in US and most of the other countries also. So if you look at the spike glycoprotein, and the center part of the Arbidol drug is going to enter and block that, and similar to the H3N2, that is influenza virus. The binding pocket is relatively conserved, and the binding positions, like 3 orbidols, can bind in the intermediate region of the protein, which can completely disrupt the spike protein trimerization. So this is a little bit hard to understand. So let's look in a movie wise, so how this works. So this is in a spike glycoprotein. So when it is in a 3-dimensional, it is in a trimer and is solid. And these monomers is all present as in a very strongly associated. So if the drug binds, the drug pocket is here. So where 3 drug binding regions are there. So you can see these, all the drug binding pocket has enough space to dock inside so that it breaks the hydrogen bonding and as well as van der Waals force of interaction. So with the aid of breaking the van der Waals force and hydrogen bonding, it loosens the structure of the spike glycoprotein and breaks the trimerization. So this abets the possible therapeutic option for a small molecule drug Arbidol to a potential possible drug for the COVID-19 infection in early entry. So another important finding which has happened, consistent with our publication, we're showing the sequence, the presence of a furin site, that is as a probability cleavage site or a furin site. It is very interesting to see none of the coronavirus, like a bat, pangolin, or previous SARS virus, has this kind of a cleavage site. This also brought in a very diplomatic and argument and controversy that whether it is a man-made virus or a naturally evolved virus, because none of the coronavirus had this kind of a cleavage site. And it has been reported very well that the presence of this cleavage site is a hallmark, means it is in a very authenticating that wherever the virus has this site, which means infection rate will be very high. So that is one of the reasons why the coronavirus infection rate is high. So now I will going to show you why this virus infection rate is high and what makes this presence of this sequence or cleavage site makes the virus more virulent or more potential or more bad. So the furin is a protease, is an enzyme which moves from a Golgi to ER membrane and also to the surface of the membrane, and where it can cleave the spike glycoprotein. So as I mentioned here, in HIV, dengue virus, and HPV infection, they can work in any place in Golgi and ER and the transmembrane, and they can cleave the spike glycoprotein. So if we may think cleaving the spike glycoprotein may be good, then what is bad? But I will show you why it is bad. So I just want to show here, it prefers in cleaving the site called RXRR, which means the basic, most of the residues which is required for its cleavage. So we saw the structure of the spike glycoprotein in complex with the furin protease. As I mentioned, this is the only site present in SARS-CoV-2 or coronavirus 2, and not present in others. And when we look at this, there are, of course, the 3 furin can bind in the 3 spike glycoprotein furin protease binding region. So this is a furin protease cleavage site, so which is an intermediate region of S1 and S2. So what happens is when it binds to the, when it binds to the S1 and S2, so it cleaves the protein in this region, the S1 and S2 region. So and this is an interacting region of the spike glycoprotein wing, or in a loop, or it is a spike glycoprotein is shown in the green, which is the target for an furin protease, which recognize these amino acids and cleave that portion. So let's understand in better way in a movie. Movies are always good to understand in very 3D way. So this is a spike glycoprotein, and if you can see in a surface model, which will be in a very clear to see the holistic view. And these regions are the furin binding sites here, and another one in the back side. So one furin and binds, which is followed by another furin. Come on. There you go. And which is followed by another furin. It can be simultaneously, or it could be all together. The furins can bind in the same time. So the binding of these furins will make a cleavage in this position. So once its cleavage happens, let me show you. So the binding of these proteins will make, this is a key residues involved in the furin cleavage site. So it completely gasps, it completely captures the complete region, and which makes the 3 binding trimer, which completely occupies, and it breaks any of the S1 domains. So breaking of that S1 domain, what happens is it separates the S1 and S2, which means the trimerization region and the ACE2 receptor binding region. Now there is no ACE2 binding region and there is no S1 region. So it is now the virus become immune to presence and absence of any receptor. So it become a pin. So the head is gone. So now the antibodies or the inhibitors or whatever you use for ACE2 or the antibodies for the S1 is of sort of no use, because it cleaves the S1 and S2 region and makes different appearance. Something like this, a person is wearing a Santa Claus dress, and when I want to see him, and he just stripped it and he's completely new person now. So based on this hypothesis and based on these observations, what we have solved and published in PNAS and other journals, we could draw a holistic model like a spike glycoprotein in a trimer, which is completely mature with a glycan on the outside, and it can interact with an ACE2 or CD26, but it also gets cleaves with an enzyme called furin. But is not just blocking an ACE2 or CD26 could be enough, but we also need to block a furin protease so that it cannot be cleaved in this position. So as I mentioned, like one target or blocking one target is not enough. We need to look for many other options. In many ways, you have to close the door. It's just not closing a main door is enough. We have to close a back door, front door, side door, whatever the doors are. So as some of the other group members are also mentioned, there are also some other advancements are happening, but this is not from our work. But this right-hand side is my work, but we have been scooped before I complete my work, and they have published in very premium journal Science. But it's very happy that they have come up with the publishing in the Science for the RDRP protein in complex with a drug called remdesivir. The remdesivir probably bind in these 2 regions. We predict it's going to compete with the RNA binding so that it can block the function of the RNA processing. This is another advancement which has made. And there is also another target is TMPRS, which I mentioned. So we are also currently working on how this TMPRS is going to work, because as I mentioned, CD26, ACE2, and there is another guy here is TMPRSS2, which also plays a critical role for changing the viral shape or changing the viral mode or something like that. With this, I end my short talk, and thank you. I welcome any questions if you have.
Yes. Thank you, Naveen. Very good presentation.
Thank
We
you,
appreciate
Salan.
your effort and your work on this COVID-19. Then it's a time for question. Anybody is interested, you can discuss with Dr. Naveen. Next.
Dr. Naveen, this is Jayaprakash. Well,
Hi, sir.
Yeah, our one fundamental question. They are talking about the antibody-based testing kits. So,
Okay.
In your knowledge, are they such a sensitive one, or whether any problem of false positives we can expect there because of the broad nature of antibodies in structure and other things?
Usually antibody tests are very promising, but if the mutations are keep going and they have any changes in the virus, but what antibodies they are using, that's also matters. But if it's a monoclonal antibodies, then it could be fine, but it could be expensive. So probably at this moment, PCR method is still very reliable and quick. That's what I think so.
Thank you. Thank you.
Dr. Naveen, Shubham here from University of Calcutta. Can you hear me?
Yes, I can.
Very correctly, you have mentioned to observe both the CD26 along with that ACE2. But what is your view on CD147 or Vaseeharan as a receptor?
I have never come across that CD147 or some other cell-determined receptors because there is no previous literature whether any of the coronavirus or influenza virus interacts with those CD146. So
47, CD147.
Yeah, even 147. I'm not pretty sure on that. Yeah.
Okay, thank you.
No problem.
Sir, this is Dr. Gopi from CPA College, Bodinaiku, South Tamil Nadu. Hi. Yeah, you have made a very good presentation, very good illustration. So, better way to understand the COVID-19. Is it possible to give a synergistic effect to rid over the infection?
Sorry, I did not get the point.
See, if you go for plant-based drugs, it will give some synergistic effect over the virus. So, because the virus is keep on mutation and it's changing its configuration. So, when we give the consortium of, a compound of plant drugs, that will affect on the virus. It's possible.
So one thing I would like to mention, I'm not denying there are no mutations. There are many SNPs or point mutations are happening, but those mutations are not completely changed the complete shape of the virus. And the plant-based drugs, I can't say no to that. Yes, definitely they all work because the drugs all start from the plant chemistry itself. But it's in a mix of the drugs, but I'm not sure whether they work or not, but we have to see. For example, you take one plant extract, there are hundreds of compounds out there. So what binds to what and where it binds, we don't know. Yeah, I'm not saying they doesn't work. They definitely probably work, but need more robust studies.
I'm asking one more question. Is
Yes,
there
sir.
any idea about working on plant-based drugs for this virus, as per your knowledge?
Probably, definitely, people are trying. I don't say no to that. People are trying that. But so, as some of the previous speakers has mentioned, it's very hard to push from the traditional knowledge of the plant-based drugs to a US FDA or Indian pharmacopoeia to level. It takes ages and many conflicts. And see, for example, many people doesn't believe in hydroxychloroquine, but some governments are pushing very hard to believe that. So it's sometimes it's hard, which is yes or no to say.
Okay. Thank you. Thank you very much.
Yeah.
Hello.
Hello.
Thank you, Naveen. Thank you for your nice presentation.
Thank you, sir.
I extend my heartfelt thank you and congratulations for your research.
Thank you.
Thank you. Jay Prakash?
Yes, sir.
Yes, next, shall we call Mahima or Swayam Banerjee?
I think we'll go with the Swayam Banerjee, sir.
Okay. Then shall we invite next Swayam Banerjee?
Yes, sir.
He's on online, I think.
Yes, sir, I am there.
Yeah, shall we start?
Yes, we can
Yeah,
start.
okay. Please share your slide and proceed, sir.
So,
Before, please, wait. Jay Prakash, please introduce about Mr. Shyam Banerjee. Shyam Banerjee. Have any profile?
No, sir. I at present I don't have it.
Okay. Sir, please introduce yourself briefly.
yes, no problem. Myself, Shubham Banerjee from University of Calcutta. I am research fellow of University of Calcutta, working in the environmental epigenomics lab. So we just worked on the molecular pathogenesis and evolutionary divergence of SARS-CoV2, and the work. Yes, and the work has been already communicated and published in the preprint version in bioRxiv as a preprint version, and the peer review process is going on. So I am going to present that work here. Can you now see my slide?
No, not yet. Mr. Banerjee, we are not seeing your slides.
If you are having problem to see my slide, I have just mailed you also my slide, so you can view that from that also. I have also mailed you my slide. I don't know why you are having problem. 1 second. Hello, can you hear me?
Yeah, we are hearing you. No problem.
I have already mailed you the slide also. If you are having problem to see my slide, you can view that. Yes, I think it is coming. Yes.
Yeah, I thought you will be sharing your screen. But
Yes,
now
I'm trying to share, but I don't know why it is problem.
No, anyway, you please go ahead. I have shared the screen.
Okay, just a full screen. Yes, thank you. So I am going to present this work, that decoding the lethal effect of SARS-CoV-2 from global perspective, its molecular pathogenesis and its evolutionary divergence. You can go to the next slide, sir. So we all know that the whole world is experiencing a new condition, that is lockdown. And our country, India, as well as China, France, Italy, those have implemented the world's largest and most restrictive mass quarantines. And I would like to add one thing in when we are talking about this lockdown, that lockdown means no social distancing, but I feel that human body distancing with strong social bonding. That is what very much needed for a current situation. So you can move to my next slide. That is my objective. So we think that the lethality of the genetic structure of different strains, we have already known from the different literature that has been already published, that SARS-CoV-2 strains are varying in different geographical locations due to some point mutations. So, but the molecular mechanism need to be understood. And besides that, the evolutionary divergence, depending upon the mutation, should also be understood. So we have tried to do that work. You can move to the next slide, please. And that is how we forwarded our work. At first, we searched for the NCBI virus database, which has the already submitted whole genome sequences of all the different viral genomes from different countries. When we started our work, that time, 13 countries have submitted their whole genome sequences from the patient's data. And we took from each country, we took one representative sequence of that viral strain from that country through multiple sequence alignment. And from there, we considered 13 sequences for 13 different countries, and then did multiple sequence alignment again using cluster W. Then we have found that different mutation positions and with that position, the nucleotide alterations that are attached. And then we constructed the phylogenetic tree to understand the evolution of those sequences. And then in-depth screening was done of those identified mutations. Then we also observed the pairwise sequencement alignment between that 2003 SARS-CoV strain and that SARS-CoV2 strain of 2020. What are the variations there? And also we did pairwise alignment with that known ancestor sequence of Wuhan, which was the first sequence to be submitted as the viral sequence of SARS-CoV2 in the database. Then we tried to observe the effect of those mutations at the protein level and then categorize the mutations as synonymous, non-synonymous, or if any deletion mutations are there. Then we did the functional prediction of those special non-synonymous mutations, and we tried to correlate the different kind of rare mutations that are observed and different non-synonymous mutations that are observed with the fatality rates. And then we tried to decipher evolutionary relationship between the ancestor one and the later developed strain in different geographical regions. You can move to the next slide, please. Yes, thank you. So in this way, our MSA analysis found total 35 positions where in the whole genome where the mutations are there. Here are two examples. You can see that in that 2269 position and the 2277 positions of the whole genome multiple sequence alignment, there is one alteration of A to T in case of Italy. In case of Italy, there is a A to T mutation in the 2269 position. All the rest genotypes are same, but only in case of Italy the mutation is there. Besides that, in 2277, another mutation is there, which is observed in case of the Indian strain, but that is not observed in other countries. The same way, one mutation is observed at the 26144 position. If you see that in that specific position, out of the 13 different strains, four different strains, including Italy, including Sweden, Australia, Brazil, they are showing the minor allele, which is T, and in place of G. So this way, we totally analyzed the whole genome, and we have found that 35 positions are there where some wild type sequence is something, and some specific strains are showing specific mutated allele. Thank you. Next, go to the next slide. So then we constructed the phylogenetic tree from those different sequence of those different countries, and you can see that this Taiwan is in the middle of that phylogenetic tree, and all the other strains are developed later on due to mutation in this way. Then what we did a pairwise sequence alignment of this Taiwan sequence with that of Wuhan sequence, which is known as an ancestor sequence of this SARS-CoV2. And interestingly, we have found that this Taiwan sequence is 100% identical with that ancestor Wuhan sequence, which established that our analysis till now is correct. Next slide, please. These are this slide showing the one part of that SMP database which we have formed. Total 35 mutations are there. But here you can see only 18 positions, but you can understand what I want to say or what we have got, that these are the wild type residue and these are the mutated residue in the position, and you can see the conservation probability of the wild type, and you can also see the variation type. And we have found, and what we did a loss ratio calculation based off those alterations to find out that which are the common mutations or very common mutations inside this specific SARS-CoV-2 genome, and we also tried to observe which mutations are very rarely observed in this specific SARS-CoV-2 genomes. And there you can see that C2T mutation out of 35 positions, we have found more than 20 are C2T mutations. So C2T mutations is very common in this specific SARS-CoV-2 genome. And if you see the loss ratio, the most rare mutations are A2T, T2A, and G2A. These three mutations are found very rare in this specific SARS-CoV-2 genome analysis. Next slide, please. So here you can see that we have highlighted that strain which are carrying the very rare mutation. You can see that Italy, there are only three mutations from the wild type sequence. Here we have shown the table in the increasing order or ascending order of achieving mutation by the strain. You can see that if we consider that Wuhan strain or the Taiwan strain to be the ancestor strain, because there is no mutation observed and total sequence is carrying the wild type genotype, then you can see that Nepal, Finland, Vietnam are carrying one mutation. Then you can see that USA carrying two mutations. You can see that Italy carrying three mutations. You can see that China, the later strain developed in China, the second strain that later developed in China, carrying 5 mutations. In India, our strain in our country, the India, there are carrying 6 mutations, and both Sweden and Spain, they are carrying 7 mutations. And the interesting findings, you can see that in the previous slide, we mentioned that A2T, G2A, and T2A, these three are the rarest mutations observed in case of specific SARS-CoV-2 genomes. And if you see, you will find that Italy is carrying 2 of those 3 very rare mutations. You can see that Spain is also carrying that T2A mutation, which is very rare mutation in this SARS-CoV-2 genomes. And if you see that G2A mutation, which is also very rare, is carried by the Sweden. And we all know that the fatality rate or the mortality rate, whichever you observe, the fatality and mortality rate of Italy and Spain, as well as also Sweden, especially Italy and Spain, the mortality rate or fatality rate is very high. And both of them are carrying this specific very rare mutation in their genome. Please move to the next slide. Then we tried to observe those mutations in protein level. We tried to find out that we all know that in this viral genome, there are surface glycoprotein or spike protein that already my previous speaker has discussed, that this spike protein is the main hotspot that binds with that ACE2 receptor or CD26 receptors or CD147 Vaseeharan receptor. This spike glycoprotein is the main viral protein that binds with the receptors in our cell, human host. So along with that spike glycoprotein, there are WRF1AB polyprotein, there are nucleocapsid protein, and there are envelope protein, etc. there in that viral genome. So we tried to observe. We tried to observe that the mutation which observed in the genome level. We tried to observe their alteration in the protein structure, and we also observed that in specific which protein this structure alteration is going to happen. And you can find that in the rightest column we have mentioned the name of the protein, and here we have in the fifth column we have mentioned the effect of the mutation in the protein level. How some mutation, you can see that in case of India, there are two mutations in spike protein or surface glycoprotein. One is that TTA deletion, which is ultimately resulting in tyrosine deletion at 154 position, and along with that you can also see that in Indian strain there is another non-synonymous mutation at 417 position. It is altering from arginine to isoleucine in case of Indian strain. So the Indian strain is carrying among 6 mutations, 2 mutations are very important mutations that are observed in this spike protein that binds with that ACE2 receptor. Along with that, you can see that in Australia also there is a mutation in that surface glycoprotein, and also in case of Finland also there is a mutation in the surface glycoprotein. So in those countries like India, Australia, Finland, we have found that non-synonymous or deletion mutations like important mutations are there in these countries. And the interesting fact is that the mortality rate or fatality rate is very much lower in these countries like Australia, India or Finland or Israel compared to the countries like Italy, Spain, etc. So this is another important observation that we have found. So this way we try to correlate that mutation in the protein level and specific position in the protein and the alteration and their correlation with the fatality rate. Next slide, please. So these are the most highly affected countries where the mortality rate and the fatality rate are very high. You can see that as I discussed earlier, the first it comes Italy. In Italy, there is that 2 very rare mutations. One is A2T at 2269 position, and another is G2A at 11,083 position. And in case of Spain also, you can see that very rare mutation T2A is observed in the position 9477. And that interesting fact is that all those rare mutations are observed in case of the ORF1ab polyprotein in case of Italy also and in case of Spain also. Next slide, please. This is Sweden. The interesting fact about the Sweden strain is that the Sweden is carrying a very rare mutation G2A in the ORF1ab polyprotein, which makes it similar to that of Italy and Spain, because we observe that same very rare mutation is observed in Italy and Spain also, and the same mutation is there in the ORF1ab polyprotein of Sweden. But we know that the lethality or the fatality mortality rate of Sweden is a little bit lower than Italy and Spain, and it may be the reason because the Sweden is also carrying a mutation in the surface glycoprotein. You know that I just mentioned while I was discussing about the strain of India and Australia and Finland, where the fatality rate is a little bit low. In those strain, the mutation in surface glycoprotein is there. So Sweden is carrying both the mutation in surface glycoprotein and also both the mutation in ORF1ab polyprotein. So this is a mixed observation from Sweden. Maybe due to that reason, it is carrying a very rare mutation till the fatality rate is comparatively lower than Italy and Spain because it is carrying a mutation in the surface glycoprotein like India, Australia, Finland, Israel, etc. Please move to the next slide. These are the mutations, main mutations we observed, and we emphasized on the spike protein or surface glycoprotein. You can see that specifically this is the when you see the detailed structure of surface glycoprotein, we know that in surface glycoprotein there are main two domains are there. One is S1 domain and another is S2 domain. S1 domain is responsible for the recognition and binding with the ACE2 receptor in the host body. So you can see that I mentioned that the countries like India, Australia, Israel, they are carrying mutations in the S1 domain of spike protein. And you can specifically see that in S1 domain, this red colored region is the receptor binding domain that binds to the ACE2 receptor directly. And the entity region is that domain which helps to bind the viral spike protein with the ACE2 receptor. So interestingly, you can see that in the Indian strain, there is a Y154 deletion and there is an important non-synonymous mutation arginine to isolation in the receptor binding domain of S1 domain in the spike protein. And also you can see that this S256R mutation, this is observed in Australia, and another 606 non-synonymous mutations is observed in case of Israel. So all those mutations are observed in the countries like India, Australia, Finland, Israel, and these are observed in the receptor binding domain, and specifically the Indian strain is carrying two mutations in the receptor binding domain of or the S1 domain of this spike protein. So we all know that if the important non-synonymous or deletion mutations in the receptor binding domain is there, then it possibly alters the structure of the receptor binding domain. So we are assuming that due to this specific non-synonymous or deletion mutations at the receptor binding domain, the structure of the receptor binding domain or the overall S1 domain is altering, and if the structure alters, then definitely the binding affinity with the ACE2 receptors and the binding interactive residues of the surface glycoprotein will definitely alter. And we think that we already know that this nature of this specific SARS-CoV2 virus binds with the ACE2 receptor with a 10 times higher binding affinity comparing with that earlier SARS-CoV viral strain. So we know that binding affinity between ACE2 and SARS-CoV2 is very high. So in the cases of India, Australia, Israel, Finland, as there are structural alterations due to these mutations in the receptor binding domain, we are hoping that definitely the binding affinity with ACE2 receptors and the interactive residues with the ACE2 receptors will alter, and that is maybe causing the less severity or less fatality among those countries. Next slide, please. So, observing the nature and tendency to achieve the mutations in specific SARS-CoV-2 genomes, we try to categorize and show the possible evolutionary way of this virus. We already know that some previous literatures have already mentioned that there are at least 2 to 3 different kind of strains all over the world. The strains has been mentioned as S and L by China and by some different researchers of different countries also. And if you go through the recent publications, there are some recent publications specifically in PNAS, the researchers have mentioned that there are possibly 3 strains are there along with S and L. And from our analysis, we are observing the possibility to be at 4 to 5 different strains which have developed from the ancestor strain of Wuhan or Taiwan through mutations. So according to our assumption, which we have got through our bioinformatics analysis of the whole genome, that from the ancestor strain, there is an intermediate strain which is developing through a common mutation, C to T. We all know that C to T transition is a very common transition, and specifically in this SARS-CoV-2 genome also, C to T is the most common mutation. So at first, all the viral strains are achieving from the ancestor strain, they are achieving this very common mutation C to T, and they are coming and developing an intermediate strain. And from that intermediate strain, 3 different strains are being developed. One is the already known lethal strain, and that lethal strain is developing further 2-3 more mutations over that intermediate strain, and along with that, 2 another different strains are also being developed. What we have mentioned as the extremely lethal strain, naming E. So you can see that the strain of Italy and Spain and Sweden, they are harboring this extremely lethal mutation like A2T or T2A or G2A, and their fatality rate and the mortality rate is also very high. And they are in green. You can see that we have mentioned that as a non-lethal strain, which is harboring favorable mutation at the viral surface glycoprotein or spike protein, which are observed in case of Australia, India, Finland, Israel, etc. And like I mentioned previously, that the Sweden is harboring both the extreme lethal, both the very rare mutation and also the favorable mutation. So maybe that is the reason why Sweden is showing a fatality rate or mortality rate in between that extreme lethal strain and non-lethal strain. So you can move to the next slide. So these are the conclusive headlines that we have found, and we can mention here that C2T is the most commonly observed mutation in these SARS-CoV-2 viral genomes, and A2T, T2A and G2A are the observed most rarely mutations in SARS-CoV-2. And the next thing that is almost clear that the fatality outcome is depending on 3 important factors, along with the factors like average age of the population or the socio-economic condition of the population. These are responsible. We know. We also agree that the socio-economic condition along with the average mean age of the population is responsible. But along with those, in molecular level, the important factors are number of mutation, rarity of the allelic variation, and the functional consequence of the mutation and protein level at protein level. And depending on that, the molecular divergence is shown that it is developing through an ancestor strain leading to extremely lethal, lethal or non-lethal strain with the involvement of an intermediate strain. Next slide, please. So what is the immediate future task? We have already started our work in these factors that we are observing the effect of those above mentioned mutations at spike glycoprotein at the structural level. So we have already developed the, we have already checked the structural alteration at protein level due to those mutations I mentioned previously, and we are doing the protein-protein molecular docking to see that due to this mutation and the structural alteration, which kind of alteration in interface area, interactive residues, and binding energy is happening. So we have already found some interesting observation in the alteration of protein structure and protein-protein interaction with those receptor proteins like ACE2, and this work is still going on. So I am not going in details with that, but we are already started to find out some important observation in structural level and molecular and the protein-protein interaction level due to those mutations I just mentioned. Next slide, please. So I would like to acknowledge my co-authors. They are Srinjana Dhar, Dr. Sundip Bhattacharjee and Dr. Pitha Bhattacharjee, and special thanks to Dr. Pitha Bhattacharjee for being such a supportive and encouraging mentor. I would like to also acknowledge University of Calcutta and UGC DAE CSR Kolkata Center for providing me the infrastructure and environment for research. Thank you. Any question, please?
Thank you, Mr. Banerjee. It's a really wonderful lecture. You have done very nice work. And from your presentation, I learned that there are three or four different kind of mutations: lethal, non-lethal, and so intermediate category. So, with reference to Indian strain, whether do you link these mutations? I think you also mentioned some deletion mutation also has happened. Can you relate this thing with the low spread of the disease? Because we have nearly, we first reported in January 29, and right now it is only 12,000 to 13,000. So do you relate this slow spread with the virulence of the virus?
Yes, actually we are assuming that this Indian strain is a little bit less lethal. You can see that if you go through the spreadability, if you go through the fatality rate or mortality rate, and if you compare that with the countries like Italy, Spain, or France, you can see that the viral strain which is spreading in India, the spreadability and the lethality is little bit less comparatively. So we have found that the both mutations are there in spike glycoprotein. In Indian strain, there are two important mutations in the receptor binding domain of the spike glycoprotein. One is deletion mutation and one is important non-synonymous mutation where arginine is converting into isoleucine. So these two important mutations will surely alter the structure of the receptor binding domain of Indian strain, and it will lead to different kind of interaction with the ACE2 receptor.
Thank you.
Hello.
Thank you.
Hello.
Yes, sir. Yes, sir.
Yes, I can hear you.
Sir, very good presentation. I want to know one more thing. That is, could you explain why Americans are more affected by this coronavirus? Because you are not mentioning that the strain is lethal for them, but we have more number of patients are dying in America. Could you explain why?
Sir, if you check the mortality rate or fatality rate in America, the mortality or fatality rate is lesser than Italy or Spain or France, because in America there have been already more than 620,000 people are reported with coronavirus positive, but their death is just a little bit higher than Italy or Spain or France, where the same number of death is observed in Italy, Spain or France with only 150,000 to 170,000 corona positive result. So if you go through the mortality fatality rate, where it is 12 to 13% in case of Italy or Spain, that is 4 to 4.5% in case of America. So in case of America, the mortality fatality rate is 1/3 of that of Italy, Spain, or France. So maybe it may be the reason that the spreadability of the virus is high, the infectivity of that virus is high, but the fatality or mortality rate is lower because no very rare mutations which I just mentioned in my presentation, that T2A, G2A, or A2T, these very rare mutations are not observed in case of USA strain, but some different mutations is observed in case of USA. So maybe we have to analyze it more and in depth to understand that why the spreadability of USA is very high. We need to concentrate on that.
Okay, thank you very much. So you are saying that the social distancing is very much important than the viral strain nature. Okay, thank you, sir.
Okay, thank you very much, Banerjee.
Hello. Yes.
Yeah, your presentation is very nice. We are getting very depth knowledge about the mutation of virus, as well as what is the strategy we find out for the next research on these mutation studies. Yeah,
thank you.
We appreciate your depth knowledge on this field. Thank you very much for your presentation. Dr. Jayaprakash Vel?
Yes, sir.
Shall we invite Mahima once again?
Yeah, yes, sir. Yes, sir.
Yeah, she's online.
I'm in line, sir.
Yeah, okay. Please be connect.
I have connected.
Yeah, okay. Please share. Please, if it is possible, give the introduction about Mahima.
সবাই তো ভেরি নাইস এক্সেলেন্ট প্রেজেন্টেশন বললো।
প্রেজেন্টেশন ভালো খালি প্রথম দিকটা তুই ওই যা করছিলি টেনশন একটু পড়ে
এরকম ভাবে ভিডিও কনফারেন্সে সবাই বলছে ভেরি নাইস এক্সেলেন্ট প্রেজেন্টেশন ইন ডেপথ নলেজ অ্যাবাউট দ্য মিউটেশন।
এত ডেপথ তো তুই পড়েই না।
না, তবে আমার আগের ছেলেটা অস্ট্রেলিয়ার ছেলেটা খুব ভালো প্রেজেন্টেশন ওর অলরেডি মানে ও আর আমি
হ্যালো বানার্জি প্লিজ মিউট ইউর
সব গল্প করছি।
হ্যালো,
ওটা খুব ইন্টারেস্টিং করে বলেছে ওর বলার ভয়েসটা খুব সুন্দর হবে।
Hello Banerjee, please mute your phone. Okay.
Good afternoon to one and all present here.
Yes.
I'm Dr. Mahima, College of Arts and Science, Rasipuram. I'm going to talk about coronavirus.
Class still on, please switch it off.
So audio is on only, sir. Hello.
Madam, you please go ahead, madam.
Yeah, proceed. Go ahead.
Hello.
Please go ahead, madam.
Okay, sir. I'm going to talk about prevention of COVID-19. Coronaviruses are family of RNA viruses, medium-sized, enveloping a positive standard RNA. They are significant viral pathogens in humans and animals. The name corona comes from crown-like appearance of virus. 4 classifications of coronaviruses are available. They are alpha, beta, respiratory tract as well as gastrointestinal infections. Gastrointestinal symptoms including diarrhea. When mutation occur, coronaviruses can be significant cause of human. There are 6. Next slide. The next slide, please. Next. The next slide.
We change the slide, madam. You please go ahead.
Next slide, sir. Human corona. So next slide.
Next slide only, madam. That is the only slide we are showing.
So next, after this. Human coronavirus have 6 types. They can infect people, alpha, beta, and beta, which has 5 types. Alpha viruses, which causes common cold. The remaining bronchiolitis as well as common cold respiratory infection. Next, in 2002 to 2003, which transmits from bats to humans, began in February 2003. So, which causes which affect the next. The next slide, please. Here, the bat coronavirus, which causes the infection transmission through animals as well as humans through diet. The next slide. Well, outbreak in Saudi Arabia. Beta coronavirus causes MERS transmitted from camels to humans. More than 2,400 cases was affected. 858 cases died. So this slide, which shows the direct history of the next slide. History of COVID-19. Outbreak of pneumonia without a clear cause in the city of Wuhan in China. A novel strain of coronavirus was detected in December 2019. They are the epidemically linked to a seafood market in Wuhan, Hubei province, China. Clinically, they are very similar to those of viral pneumonia. The initial symptoms are fever, cough, fatigue, and similar to the SARS. Next. World Health. The next slide, please. World Health Organization. The disease COVID-19 on 30th January 2020. This is of virus named the virus severe acute respiratory syndrome coronavirus 2 SARS-CoV2. So far, the disease has rapidly spread from Wuhan to China and other areas. Next slide, please. Mode of transmission. The virus is highly infectious and it's transmitted through droplets and contacts. Some patients are life-threatening and disease has caused a great threat to global health and safety. So the control of the spread of the epidemic and reduce the mortality as soon as possible. To reduce the morbidity and mortality of this disease, as pandemic. The diagnosis and treatment of this disease with the RTP derivative hydroxychloroquine along with several other antiviral drugs would be effective strategy to count COVID-19. Chloroquine is exported from India to various countries for the next slide, please. Next slide, please. India's first COVID testing kit which was produced by when she was pregnant. Next day there was delivery, even though she detected the patho detect kit. So it is used to detect rupees within 2.5 hours. We can detect the coronaviral infections. In other countries, they are taking
7 to 7.5 hours. Next slide, please. The next slide, please. There are no vaccine and specific drugs available. Ayush recommend homeopathy and Unani medicines, the core such as drinking Shadanga Paniya, Agastya, Hartyagi, and other Unani drugs. Kabasura Kudinir, a product of COVID-19 infection. Then Thiruchengode used to treat sore throat and cough. Next slide, please. The next slide, please. Plasma therapy would be mostly the patients with renal workload and help to promote the recovery of renal function. Vitamin C may prevent the susceptibility of lower respiratory tract infection under certain conditions. Ayush recommends persistence of coronaviruses. Next slide, please. The coronavirus is which persistence with absorbed aluminium 2 to 8 hours, surgical gloves 8 hours, steel 48 hours, 4 days, glass 4 days, paper 4 to 5 days, plastic 5 days. Next slide, please. So, COVID-19, the organism, the virus, next slide, please. The virus scientific studies have proven that apart from the nasal and respiratory secretions, the COVID-19 virus is actually shed in the stools of infected patients. This shedding may continue to occur for
Okay, ma'am.
Shall I continue? Okay. So these are the symptoms of 2019 coronavirus: fever, chest pain, chills, rapid heartbeat, breathing difficulties, pneumonia, kidney failure. Finally, it leads to death. Next slide, please. In severe cases of, next slide, please. In severe cases of pneumonia, system immunity plays an important role in maintaining optimal health. We all know the prevention is better than cure. Where there is no medicine for COVID-19 as of now, it will be good to take preventive measures which boost our immunity in these times. Ayurveda being fine, healthy and happy. Next slide, please. In Ayurveda, they are giving the concept Dinacharya, a daily regime. As well as Ritucharya, seasonal regime to maintain healthy life. It is main a plant-based is emphasized across Ayurveda classical scriptures. For individuals, taking protective measures can effectively prevent SARS-CoV2 infections. Improving personal hygiene, wearing medical mask, adequate rest, and good ventilations. Next slide, please. Ministry of Oil's recommendation based on Ayurvedic literatures: drink warm water throughout the daily practice yogasana, pranayama, and meditation at least for 30 minutes. Spices like turmeric, immunity enhanced diabetes should not should take them every 7 days. Drink made from basil, tulsi, cinnamon, Ayurvedic procedures, nasal applications. When we can apply the sesame oil or coconut oil or ghee in both the nostrils, the epithelial layer which prevent the in the nose, so it may prevent the infections. Oil pulling therapy is a day. Then dry cough can be treated with dry cough or and sore throat can be treated with pudina mint leaf for steam inhalation or a neem taken 2 or 3 times per day, which prevents cough or throat irritation. So these measures normally we can take. If more symptoms persist, we can go for physician. Next slide, please. This is the Ministry of Ayush told whatever the ingredients which is normally available in our home, we can take these materials so we can cure the infections. Next slide, please. So here, this is also one kind of boosting immunity. This is also given by the Ayush. Here also, you can do different kinds of meditations are available that also which improve our immune system. Next slide, please. Social distancing and washing your hands frequently with detergents or sand. Next slide, please. Here, we have to maintain 1 meter distance in hospitals, malls, marketplaces, restaurants, and other places. Work from home, avoid meetings, classes, tutorials, workshops. We should not go for crowded place or public place. We should not go for social or other meetings or events. We should not shake our hands, and we should avoid non-essential travel and tourist trips. Next slide, please. Wisdom is not about education. Responsibility has nothing to do with status or affluence. This photo from a tribal village in interior Odisha. Here they are maintaining the distance, social distance. Next slide. Wash our hands, stay at home, use medical mask and sanitizer, avoid contact with the sick people, or avoid slide, please.
Your voice is breaking. Make it fast, please.
Yes, sir. Okay, sir.
Or skip a few slides then.
Prevention of COVID-19, wash your hands, stay at home. We should not omit those rules. So in India, as well as given by CEO, American CEO, in Italy 60,694, Spain 58. But in 30th of March, in India 1,328. But in United States 1,43,000 something people, in Spain 85 something, and 85,000 something, Italy 97,000 something. So we have to maintain the quarantine. Sleep early and wake up early. Brush your teeth and gargle with warm water. Ginger, cardamom, tulsi, mint tea boost our energy. Make yourself busy with some hobbies. Next slide. Hobbies, be happy, stress free, no stress. Next slide, please. Next slide, please. Okay. So here, sunlight is a gift from nature. Surya Namaskar, we have to do blue light from sun, increase blood circulation, helps to detox. Vitamin D gives better sleep. Next slide, please. Yoga. Next slide, please. Following Surya Namaskar. Following Surya Namaskar. Simple breathing exercise strengthen our respiratory system. Next slide, please. Moon bathing for 30. Next slide, please. Oh, moon bathing for 30 minutes. Get away from artificial light. Observe moon's cool, refreshing light. Observing our energy. Observe and next slide, please. Solution to treat COVID-19 through Ayurveda is near future. In India, we are having tremendous plans. Antiviral, antiparasitic effects. So next slide, please. Next slide. So role of Ayush. Yesterday evening, role of the Ministry of Ayush, Government of India. So take Ayurvedic home remedies and boost your immune system. Stay home, stay safe, stay alive. Thank you. God bless you today and always. Thank you, sir. Thank you for giving me the opportunity.
Thank you. Thank you, Jay Prakash. Shall we invite the next presenter, Dr. Jnanendra? Dr. Jnanendra?
Sir, I have one question, sir.
Yeah, please.
Hello, sir. Sir, actually, I'm Manikandan from Kalisari College, sir. First of all, I thank to Mahima madam to share this knowledge to us. Recently, America bought hydroxychloroquine from India. Actually, we know about hydroxychloroquine is used for prevent. Madam, are you here?
Yes, sir. I am here now.
Madam, actually, recently America bought hydroxychloroquine from India, madam. Actually, we know about hydroxychloroquine is used to treat malaria caused by mosquito bite. Is this any prevent activity against coronavirus, madam?
Sorry, sir.
What is your opinion?
Sir, please repeat the question due to network problem.
Madam, actually, recently America bought hydroxychloroquine from India, madam. Madam, are you listening? Hello, madam.
Sir, please type in the chat box so that she can read.
Yeah.
Okay.
Sir, please.
How malarial drug hydroxy is helpful in corona treatment? He is asking.
Yes, madam. Yes, madam.
Actually, I'm not medicine practitioner. They are all given. I think so. They have given so many drugs for the treatment of corona
Oh, okay, madam.
COVID-19 patients. This drug which
Hello, madam. Sorry, your voice is break, madam.
She has her poor net
somewhat. That's why they are using this drug. Further researchers.
Okay, thank you, Makima, for your presentation. Sorry for the inconvenience in between. Network made some trouble for
Okay,
us.
no problem, sir. Thank you, sir. Thank you,
Okay,
sir.
thank you. Next, we invite Dr. Jnanendra.
Hello.
Yeah. Good evening, Jnanendra. How are you?
Sir, good evening, sir.
Can you please introduce him, sir, to the audience?
Yeah, please. Jay Prakash, please introduce Dr. Jnanendra, please.
Hello.
Wait a minute.
Hello.
Hello.
Yes.
Yeah, this is we are happy to introduce Dr. Jnanendra to the audience here. Dr. Jnanendra is presently working as assistant professor in the Microbial Genomics Lab, Department of Biotechnology, Yungnam University, South Korea. He has a PhD in bioinformatics, and he also holds a certificate professional program on protein modeling and rational drug designing. He has completed both MSc as well as MPhil in MSc in bioinformatics and MPhil in biotechnology. So he has been working with the Yungnam University, South Korea since 2018. Previously, he has served as postdoctoral research scientist in the Department of Biotechnology of the same university. He also worked as a research associate in the Center for Research and Development, Mahendra Engineering College, and visiting faculty at the same department during 2014 to 2016. He also worked as training consultant in the bioinformatics division, Origin Solutions, Salem, and he also worked as assistant professor and joint director for research in the Selvam Arts and Science College, Namakkal from 2012 to 13. He also worked as HOD in the Department of Bioinformatics at the Vivekananda College of Arts and Science, Thiruchengode, 2009 to 12. And again, he worked in the same college as lecturer from 2004 to 2009. He is having a vast number of publications. I'm showing the publications. More than 25 publications he is having. He is having a strong skill set in the next generation genome sequencing as well as bioinformatics expertise. So he has delivered many invited talks as well as the other things. He has attended so many conferences and published in several papers. So he is one of the organizing committee member, and we request now Dr. Jnanendra to present.
Hello.
Yes, sir.
Yeah, thank you. Thank you for an introduction, sir. Can you able to see my slides?
No, sir. Your
Please
slide
share
is not
your slide.
My screen is shared.
No.
No, you please reshare it again, sir. So I have shared your CV now.
Okay,
Yes,
okay.
sharing.
Yeah. Is it okay now?
Yes. Share your full screen.
Okay.
Okay, fine.
Okay.
Yeah, it has come, sir. You please go ahead, sir.
Yeah, my slides is visible, right?
Yes, sir. Yes, sir.
Yeah, okay, fine. So I am here to talk about mining the genome and drug targets of SARS-CoV2. And as we have seen from morning, so many people has given a nice introduction about SARS-CoV2. And just I would like to give a few points which we should remember. It's a large enveloped RNA virus which is having nearly 30,000 nucleotides. And the one special feature of this SARS-CoV2 is It's having an two kinds of an replication mechanism in which we can see an ribosome frame shift during genome translation and synthesis of multiple subgenomic RNA, so which helps in making an various parts of this genome. So let's see that how the genome of our SARS-CoV2 looks like. So this is the genome structure which shows you from 5' end to 3' end. So where we are having and two kinds of an protein regions. One will be an replicase polyproteins, and the next is of an structural protein region. So if you look into this replicase polyprotein region, we have and two ORF regions which account for almost 16 non-structural protein regions. So this ORF1A and ORF1B. So it's almost 21,000 nucleotides. It accounts for 21,000 nucleotides. And the second part of our genome is in structural proteins, which consists of almost 4 important proteins. One is of an spike, next is of an envelope, membrane, and nucleocapsid. So these are the 4 important regions which helps the virus to, it shows in different aspects. We can see what are the functions of each and every protein. And coming for this first ORF1A, so in which we are having in 2 specific regions, which is very important, like papain-like protease and pre-CL protease. And this is the structure which has been available in an PDB database for us. So 6Y2E, that's an accession code. Those who would like to work on this protein, so just we can download that protein, we can view here, and we can go for some docking studies and or what are the drugs which you would like to recommend. And next, second ORF region is of an ORF1B, which consists of an 3 important proteins. One is of an RNA-dependent RNA polymerase, and next is of an helicase, and next is of an endoribonuclease. And these are the structures which has been available. And as we have seen in previous lectures, like the spike protein, which is very essential for binding to an ACE2 receptors. So this ACE2 receptor and the spike protein interacts with a specific region called as of an RBD region seen on your spike protein. And this specific region is called as of an RBD, that's an receptor binding domain. And we can get the structure of this RBD protein too. And digging more deep into that, the second specific region, apart from a spike and membrane and a nucleocapsid, we have and 3 other regions. So the structure of E protein, which has been recently shown, the top view of that, and this is the side view of our A protein, which has been available in PDB. And this is the structure of an ORF3A, and this is a region where we are having an the no structure has been identified, and ORF3. And this is about the genome. And based on this E protein, so now currently the diagnostic kits which has been made available to detect whether the patient is positive or negative. So this is the steps which has been involved in that. So first thing is just nasopharyngeal swabs will be collected from a patient, suspected patients, and it takes almost less than 15 minutes. And once the specimens has been collected, so they can be stored in 2 to 8 degrees centigrade up to 72 hours. And then the extraction of RNA will be taken, so which will take around 45 minutes. And once the RNA has been extracted, we are going for an PCR reaction. So here the most important thing is they are using 2 different kinds of genes which has to be expressed. So the first one is of an E gene. So and this is the primer which has been used as in forward and then reverse to carry out an PCR reaction. And the second region is of an RDRP gene, and these are the 2 probes which will help us to detect the presence of these genes. So if the patient was said to be N positive, this will be the graph. So where the fluorescence will be very high, it says that the number of virions, the number of E or R proteins has been seen rich. So if the number is less, it's less than threshold, then the patient will be considered as of a negative. So this is the working mechanism of our COVID-19 diagnostic kit. So these are the 2 regions which plays a crucial part in a diagnostic kit. And coming to a brief cycle of this COVID-19, the site of infection is mainly lungs, in which we can see an alveoli. And if you look into an alveoli, so this will be the structure of our alveoli. So where when you breathe in, the oxygen comes in, and when you breathe out, the oxygen, carbon dioxide goes out. So this is the point where the virus enters into an alveoli and it sticks to an alveoli cells. This we can consider as of an alveoli cells, on which we have an ACE2 receptors, and to which the virion particle binds to. So if you can consider that this is of an epithelial alveolar cell, and this is of an receptors, ACE2 receptors, and we have our virion protein, we have our COVID-19. And so if you can look into the structure of this, these are the various proteins which has been seen on our coronavirus. So which is having an spike proteins, nucleocapsid, membrane, envelope, and an RNA viral genome. So these are the parts which has to be separately synthesized inside the cell, and then they will be assembled into one complete structure. So if you look into the structure of this virus, the spike protein is a major one which helps to bind to the ACE receptor, and this is the structure of our spike protein. So once the virus comes and attaches to your ACE receptor, so you can look into the structure of this. So this is how the closed conformation of your spike protein looks like, the closed region. And when it binds to your ACE receptor, so the domain opens. So this is the place where the changes has been occurring in your protein conformation. So this conformation is very essential for its interaction. So closed conformation and an open conformation of the protein. So those who wants to study the closed conformation of the protein, they can just download this PDB ID, and for open conformation, this is the PDB ID. And so once it's been done, you can look into that specific region of the spike protein, so which is having two specific domains. So like S1 domain and S2 domain. In an S1 domain, this is the region which has been helping. So you can see that the blue colored highlighted regions are the one which is having an interaction with an ACE2 receptor regions, and as well as, and here we have an polybasic cleavage site where O-linked glycan residues has been observed. So these are the specific regions which has been helping for binding of this S2 domain. And this is the picture which has been taken from publication of an Nature. The credit is for Nature journal and the authors who has published that. And so once the virus attaches to an ACE receptor, so it will be engulfed into the cell, and where the viral genome will be released. And this is of a single-stranded RNA genome, a plus sense. And once the viral genome has been released into the cell, the ribosomes which has been present in the host cell will be utilized for its synthesis of various proteins like viral polymerase. And this viral polymerase helps in making an other necessary proteins, and it's called as of a viral replication. So by this time you could have understood this. This is of a viral polymerase, so which is nothing but our RDRP. So RNA dependent RNA polymerase, ribosome dependent RNA polymerase. And next is of an RNA replication. So where the strands will be, the negative strand will be synthesized, and this is the mechanism where the viral protein will be synthesized. And this specific, at this specific region, it takes and two specific pathways, like producing an genomic replication, and next one is often subgenomic replication. So where we have the structural proteins has been synthesized. So once this structural proteins has been synthesized, among this spike protein, a membrane protein, and envelope protein will be translated into an structural proteins, and which in the site is often endoplasmic reticulum. So in the endoplasmic reticulum, this proteins has been synthesized, and once they have been synthesizing, while they have been synthesizing here, the nucleocapsid and genomic replicated products will be assembled. So that's of a nucleocapsid, and all these three together forms into an specific virion. Hello.
And all these three together forms into
Hello. So here this endoplasmic reticulum will be transported into an Golgi complex, and once they have been matured form, they will be made into an endoplasmic they will be made into an Golgi complex vesicles, and from the Golgi complex vesicles they will be transmitted out to the other cells, so that they are ready to attack the other subsequent cells. And this process is called as an exocytosis. So this is a complete replication mechanism. So once we can understand this mechanism and which proteins has been and which gene has been helping to make a specific protein and which protein has been playing a crucial role, so we can identify the drug regions and we can specifically target what are the drug regions. So now we know that what is a mechanism, how it has been replicating inside the cell. So now let's come for an targeting the specific regions. Like, so these are the drugs which has been proposed. So you can see here that the viral entry mechanism is like with an spike protein, so where it attaches to an angiotensin converting enzyme receptors. So here we have an specific regions like an S1 subunit and S2 subunit, where it binds to your ACE2 receptor. And now these are the two drugs like monoclonal antibodies targeting S1 or S2 regions, and these are the drug names. So which has been antiviral peptides, which has been under test to block the entry of this SARS-CoV2. So these are the potential drugs which has been used in clinical trials to act against this viral entry mechanism. And next, coming to the other different kinds of drugs which has been testing now, is like if you look into the genome of this one, we have an papain-like protease. So where we have an PL-PRO inhibitors, which has been, this is the drug which has been currently testing. And next one is of an 3CL protease, for which lopinavir has been currently suggested to inhibit its action. And next one is of an RNA-dependent RNA polymerase inhibitor, so ribavirin, and this is a drug which has been currently proposed for that. And next one is of an helicase inhibitors, and they are suggesting an siRNAs against structural and accessory genes which can act on this specific small structural proteins. So these are the other drugs, and the most importantly, we have an 1 to 16 non-structural proteins for which they have been suggesting an alispovir as an which is of an CYP inhibitors, which can act potentially on this specific non-structural proteins. And this is the mechanism how the remdesivir has been proposed and how they have been using that in order to have the potential activity against your RNA-dependent polymerase. So as soon as the virus enters inside, so this is the viral protein, so where the remdesivir drug actually acts on, so which can inhibit the RNA replication mechanism. So where we have and different the other subunit products will not be synthesized further, so that we can stop the replication mechanism inside that. So this is the one of the drug which has been currently now under and phase 3 trials. So successfully this can be, hopefully this drug can be made as of a successful antiviral drug soon. And this is the binding region where it binds to, and this is the PDB ID. If those who wants to work on this, those who want to modify the drug, and they can just work on this by using your docking studies and all. And this is the possible mechanism of an chloroquine. So once the once the virus enters into that, it is said to be having a different kinds of an side of actions. So where it may prevent the formation of clathrin complex during endocytosis. This is the first place. Or second, it may interfere with endosomal acidification and viral genome release, so which can increase the pH, so that acidification will be increased. Or the next one is may affect post translation modification and viral assembly. Or next, it may interfere with vacuole formation and viral budding. So these are the places where the chloroquine can act successfully. And apart from that, this chloroquine has been given as of a drug, not as of an antiviral drug. So it has been given as of an endosomal acidification inhibitor. So it works against an host, whereas the antiviral drug should work against any one protein of an viral. But this will be considered as of the host target, and one can successfully inhibit the assembly of an virion by using an membrane-bound RNA synthesis inhibitors. So this is about a small glimpse of study what I have made on to understand how this COVID has been, how the COVID replication has been taking place, and what are all the different drug targets we are having, and what are the two different drug targets which are in race to exist as of an potential antiviral drug targets. And that's about my talk. And thanks for giving me an opportunity. And I thank the organizing members for giving this wonderful opportunity. Thank you everyone. So if you have questions, we can discuss.
Hello. Good afternoon, Mahendra sir. I'm
Yes.
Elilars, sir.
Hello.
Hello, sir. Hello.
Yeah, hello.
Sir, the plasma therapy is more effective. Hello.
Yes.
Is there any chance to raising the anti-sera in notobiotic animal like horse and all, sir? Is there effective against corona?
Actually, there few studies has been saying that they can give an plasma therapy, but still they has to go for an FDA approval for that.
Oh.
So we don't know, but I think here in Korea they have found some few antibodies, and they are suggesting that, and they have reported that it's successfully they can treat the patients. But I don't know actually how far it has been happening.
Okay, sir. Thank you.
Sir, do you think Indians are have better immunity than other country people as far as
Actually, when the pandemic has been started, the epidemic has been started in China, we Indians has believed that we will never be targeted with this because of our immune system and all. And later, at later stage, we came to know that the virus is undergoing and various kinds of mutations, and which if we would have seen in an previous talk, he has reported wonderfully that what are all the mutations which has been seen in each and every country. So probably it is nothing to do with an immune system. That's what I feel.
Sorry if I'm wrong. Actually, we Indians got immune to all pollution and all kinds of things. That may be also one reason, no?
No, it's not like, maybe you are true, but the thing is we have never been exposed for this kind of a new virus. So you should think in an
we have not been exposed to virus, but in general, we live in a polluted place, whether water or air. So continuous pollution, we got immune. Our system has got the immunity compared to other people.
System is having immunity, but the problem here is we should have in specific antibodies which can fight against to the specific virus. If it is after few more years, definitely you will be getting an immunity towards this specific virus. So in future you will not be having this.
Yeah, even for women, they say compared to male, female are less affected because of the secretion of estrogen and progesterone. In what way it is connected to corona? Can you say?
I'm sorry, I don't have specific idea on this part.
Okay, sir.
Okay, thank you, Mahendra.
Mahendra, sir.
Hello.
Hello.
Sir, one more question, sir. One more question.
Yeah, sir.
From me.
Yeah, sir.
Sir, temperature and humidity can play a major role for against the corona. In low temperature countries like China, America.
Yeah, definitely
Iran.
we can say that the temperature and humidity plays a role, but it cannot completely stop the spreading of that virus. But the spread rate will be less.
Yes.
Spread rate will be less, but it's not going to completely eradicate the spreading.
Okay, sir. Okay, sir. Thank you.
Okay, sir.
Sir.
Hello.
Yeah, I am Dr. Gopi from Bodinayakno, CPA College.
Okay.
You made a very good presentation. It's highly illustrative. What would be the plan for India? So they are locked down up to 3 May. So after that, there are the climate will be changed. So maybe rainy season and followed by winter season. So what could be the infection rate? Even the summer season itself, it shows the vigorous. If the further season comes, so what would be the strategy? What precautions should take? So any.
So the strategy for this is simple, as everyone is saying that we should maintain an hygienic. We should have an hygienic, and then we should maintain a social distance for a while until we should have we have an specific medicine or vaccine. So it's better. And moreover, the thing is, if you look into the graph, it seems to be increasing day by day, and maybe soon after 25th or 26th of April, it may start to decline. But still, the people are worried about the situation, how it is going to be once the lockdown has been lifted.
Yes.
So, but each and every citizen should be having their own view, and they should strictly follow the rules. Otherwise, it will be same. And the people should have their they should have their own they should think that they have their own families, so that they should not spread the disease to everyone.
Actually, they should follow ethics, and they should follow all type of hygienes. At least by this, all people will follow hygiene. And season, as Dr. Gopi sir, I think, he asked, seasons also may change the severity. Sometimes heavy wind or heavy rain will flash out all these viruses also. Let us think in a positive way.
Yeah, maybe.
Hello, Gnani.
Hello.
Thank you. Hello, Gnani. This is Dr. Rajesh. Thank you. Thank you very much for your nice presentation. And I would like to ask you one thing. What would be the reason behind that mutation of these virus? So you spoke about different type of proteins and all. And in the previous lecture, he spoke about variations and all. What could be the reasons? I hope that there may be various reasons, but based on your assumption, what could be the reason behind these mutations? Whether the human being, the nature of human beings, or the climatic conditions outside the body.
All the mutations for the existence.
Yeah, it's exactly said by Selvan sir. Yeah,
Yes.
almost all the viruses tries to have an resistance towards some specific antibiotics. As we study in an bacteria, we know that each and every bacteria tries to have develop an resistance towards some specific antibiotics. In the same way, the viruses tends to have an mutations. And till now, I don't know how many has been gone through this. They have reported nearly 3,500 mutations in this COVID-19 virus. Almost 3,500 mutations. So
Yeah, that is what I'm asking. Actually, whether
it's
it is with reference to the body of the human or with reference to the climate outside the body. What could be the possible cause of this?
it could be anything. It could be an once if the virus sticks on to any specific notebook, so it tries to change. It tries to accommodate itself, so that it needs to exist at that place for some more time. So it can make some changes in its body, in its proteins, and it so that the mutation occurs. But when it comes to an human body, it is having an enough supply of an food or whatever the thing it needs from a human body. So it tends to adjust to the specific what to say nature.
Sir, good evening,
Oh,
sir. I have one doubt,
okay,
sir.
Yanni, thank you.
Thank you.
Hello, sir.
Hello.
Hello, sir. I have one doubt regarding plasma therapy, sir.
Okay.
Sir, actually, the plasma therapy, will it cure the corona disease or will it prevent the corona disease?
So it helps to fight against the coronavirus. So once the antibodies has been increased, it starts to it starts to fight against that. So gradually your immune system will increase, so
Okay,
that
sir. Okay.
slowly the virus particles will be removed out of our body. So
Okay, sir.
it's a slow process, but it will be taking around 14 days.
Oh,
As
okay,
of
okay.
now, I learned.
Okay, sir. Okay, sir. Thank you, sir. Thank you.
Okay.
Okay, thank you very much for your nice presentation, Jani.
Thank you, sir.
Okay, it's a time for end of this e-conference.
Yeah.
Dr. Jay Prakash.
Yes, sir.
Yes. Let me close the session.
Shall we close? I think Muhammad Ashad is not here.
Okay.
He, as per schedule, he is not here. He is not.
Okay.
I think he is not here.
Okay, if it is not reported, we can go for ending the session. Anybody have give any good comments? We can encourage
Yes,
for
sir.
just 5 minutes.
Some
Please give
more
feedback about. I already mentioned. If it is any feedback, you have to send to our mail ID biotechnology@gmail.com. I am sincerely thanks to all the participants for your kind cooperation and for the successful fulfillment of this e-conference on today. Is anybody have to give?
Sir, thank you very much. Very good attempt, sir. I think
Thank
you
you,
can
ma'am.
do more like this till the lockdown period.
Okay.
First itself is very successful, so and all the presenters did very well.
Okay.
Thank you so much
Thank you,
for
ma'am.
the
Ma'am,
opportunity. I'm Dr. S. Parvati, working as assistant professor in Government Arts College, Salem.
Okay. Thank you, ma'am.
Sir, I'm Dr. G. Manigandan from Kalisari College, sir. This is my
Okay.
first e-conference, sir. First of all,
Oh.
I thank the Biological Science Forum committee members, and also I thanked guest for delivering his lecture, sir. This conference is very useful for me and other participants, sir. Once again,
Thank you.
thanks, sir.
Thank you, sir.
Thank you.
Selvan sir,
Yes.
congratulations for the successful completion of this conference. And I thank all the presenters who have presented here today. They have done a very good work. Even what I feel is now you can compile all that presentations, and you can with the prior permission from the presenters, you can make it into a souvenir so that it would be very helpful for any researcher. Even some of the sessions in the last, like Jnani session and previously Mahima madam, they have given some initial ideas to begin with that, and advanced lectures also been presented here. So I think you can compile all those things, and it would be a very good research material for any researcher who want to work on COVID. I congratulate you and your team for the successfully organizing this thing, sir. Thank you very much.
Thank you, Rajesh.
Hello, sir.
Yes.
This is Dr. Mahendra Kumar from Guru Nanak College.
Thank you, sir. Welcome, sir.
The conduct of the conference was very much planned and very well executed, sir. For that, on behalf of our college, I would like to thank the organizers, especially Selvankumar sir and Jay Prakash sir. It was more like actual conference, and all the PPTs sessions were really informative, and it really cleared many of our doubts, sir. Thank you very much for that, sir. And thanks for the opportunity you gave to many of us for attending this conference.
Welcome, sir.
Sir, this is Dr. Gopi from CPA College, Bodi Naikanur, Theni district. Really, it's a wonderful arrangement of e-conference. I think from the beginning itself, there are about 100 participants. It's really interesting. It's a highly appreciated one. So, as I said, one of the participants, you better to, prepare a souvenir, and also certificate. So it will be useful for the career. So thank you, one and all the organizers. Thank you very much.
Thank you, sir.
Hello.
Yes, sir. Saravana, sir.
Yes, sir. So it's very good, informative about the COVID-19. Really helpful for us, young budding scientists. They are more interested in this regard, e-conferences. Thank you for organizers for good organizing process. Thank you. Thank you.
Thank you, sir.
Sir, this is Dr. Srinandini Devi from Kaidamilath Government College, Chennai.
Welcome.
Thank you for the organization and congratulations to our team. Sir, along with me, even my son, who is doing 6th standard, he also got to know many things about this virus. He really understands the situation seriousness, sir. Thank you so much for this opportunity given, sir, once again. Thank you.
Sir, Madhavan, sir, you have any feedback, sir?
Professor, please.
மிகவும் சிறப்பான முறையில் ஏற்பாடு செய்த
Okay, shall we?
Hi, sir. I am Arthi Subramanian from SSN College, Biomedical Final Year.
சிறப்பான முறையில் ஏற்பாடு செய்த
Thank you for your wonderful session.
என்னுடைய மனமார்ந்த பாராட்டுக்கள்.
Please, anyone keep on mic.
Sir, good evening. Good evening to one and all present here. So I am Dr. Mahima from Muthemal College of Arts and Science. On behalf of myself and Muthemal College of Arts and Science, I want to thank Dr. Selvan sir and one more sir from Chennai, organizers. Then on behalf of myself, this is a very excellent biological e-conference. This is I think this is the first time we have organized such kind of so many resource persons and so many participants. I congratulate the resource persons, organizers, and participants. Thank you very much.
Okay. Next, this program they conclude with the vote of thanks. I would like to thank Professor Vaseeharan sir for give some introductory talk, and Dr. N. Madhivan, Professor and Head Department Director CS in Botany, University of Madras. He has given wonderful talk. He is also patiently with us from the end session. I feel he is one of his father like that. For our entire guidance and promoting to us for the next level. I am also thank to the Grinath Pillai. He is the hero of this session because he provided for this host facilities for this Google Meet for us to connect more than 250 people. So I especially thank to Dr. Grinath Pillai and his organization Nairo Research in India. We are also patiently cooperate with you, sir, in future. Then special thank to VLS Professor Department of Biotechnology. He was given wonderful talk about COVID-19 till the current data, everything. Then Dr. N. Mahima, Assistant Professor Department of Microbiology, Muthema College. Then one of my student, Dr. V. Naveen, Research Fellow, Monash Biomedical Institute, Australia. Then Mrs. Dr. V. Gomadi Arunan, Independent Researcher, Chennai. Then Dr. Niyas Agamat, Assistant Professor, Department of Biochemistry, Faculty or College, Tirupattur. Then Swam Banerjee from Environmental Epigenetics Laboratory, University of Calcutta. Then my mate friend, Dr. Jananda for me the technical session as well as online. Thank you to Organizing Secretary Dr. J. Prakash Vel on behalf of this Biologic Science Forum. Your offer is very nice, Dr. J. Prakash. Thank you to all the participants for this biological science forum concern. Thank you, one and all. Jay Prakash?
Sir.
Anything?
Nothing, sir. I also, on behalf of the organizing committee, thank you profusely all the speakers as well as all the participants. And I especially would like to thank you for muting this idea. It is the brainchild of Professor Selvankumar only. He has only promoted this idea and organized all the people. He gathered most of his students itself for the meeting. Initially, when he was proposing, I was thinking that we will be having somewhere 30 to 50 participants. But because of his wide contacts, he managed to get more than 200 registrations. And we have seen in the morning 150 registered participants have joined in the meeting. And till now, we could say that we could see around 100 people are still joining with here. And I think this is the conference, only conference where the speakers were not urged to complete the sessions. And all the interactions were allowed without any hesitation. All the people were allowed to have enough time. And we originally planned to complete the sessions by 3 o'clock, and now it is 4 o'clock, just 1 hour only.
Not even lunch time, sir.
Yes, yes.
It is a nicely organized conference, sir.
Everything is on service motive.
And the team. I thank you once again, sir, all of you. And I thank you so much, particularly Professor Selvankumar. Thank you so much, sir.
Thank you very much for all the participants and presenters, and provide the support by the Grinath Pillai and Janendra, please. Thank you very much.
Yes, sir.
Dr. Grinath, are you here? Okay, let's close the session, please. We will send the certificate within a week to your registered mail ID. If you are not supposed to register in mail, please send to your mail, send our biological email.com. Thank you. Sir, thank you, sir. Sir, Madhivanan sir is there? Good evening, Jani.
Sir? அல்ல அது லிங்க்ல இருந்து தான் வரணும்